participants and attendees of the first international conference on problems and perspectives of modern science. Me, Zamonaviy, professor of the Department of Physics and Chemistry at the Tashkent Institute of Irrigation and Agricultural Mechanization Engineers, acting as a moderator in here today. I'm very pleased to welcome you all to this exciting upcoming event that will unite us all for one common goal. I have one special request. We kickstart the opening ceremony. That would be kind of you all to keep all the microphones off and the video cameras on during the event. As the opening ceremony of this conference is just a few seconds away, I wish you each enjoyment of the events to come within the next few hours. Once more, good afternoon, ladies and gentlemen. Here that the conference has already begun. I want to inform you that for the opening part, we have planned about 1 hour time slot to deliver the opening speech. I invite the rector of the Tashkent Institute of Irrigation and Agricultural Mechanization Engineers and head of the Higher Education Council, Professor Uktam Umurzakov.
Assalomu alaykum. Katta rahmat moderatorga. Hurmatli xalqaro anjuman qatnashchilari, muhtaram hamkasblar, mehmonlar, xonimlar va janoblar, avvalambor barchalaringizni IQXMMI 2021, ya'ni ilm-fanning zamonaviy muammolari va istiqbollari mavzusidagi birinchi xalqaro anjumanning ochilishi bilan chin dildan tabriklayman. Bizning Toshkent irrigatsiya va qishloq xo'jaligini mexanizatsiyalash instituti tashabbusi bilan Janubiy Koreyaning Sunmun, Navoiy davlat pedagogika instituti, Toshkent davlat transport universiteti hamda Toshkent kimyo texnologiyalar ilmiy tadqiqot institutlari bilan hamkorlikda tashkil etilgan ushbu xalqaro anjuman respublikamizning ilmiy amaliy yo'nalishdagi istiqboli uchun muhim qadam hisoblanadi deb umid qilaman. Konferensiya dasturidan ko'rganlaringiz yoki ko'rishingiz mumkin. Ushbu xalqaro anjuman ilm-fanning turli yo'nalishlarini qamrab olib, qishloq va suv xo'jaligida innovatsion usullar, ilg'or kimyoviy texnologiyalar, bino va inshootlar qurilishi, injiniringi, energetika va sohalar ilm-fanida raqamli texnologiyalarni qo'llash bo'yicha yetakchi olimlarning ilmiy natijalarini qamrab olib, keng ilmiy munozaralar maydonini shakllanishiga xizmat qiladi degan umiddaman. Ilm-fanning zamonaviy muammolari va istiqbollari mavzusidagi birinchi xalqaro anjuman tashkiliy qo'mitasi rais sifatida ishonchim komilki, anjuman yo'nalishida tadqiqotlar olib borayotgan va bevosita ishtirok etayotganlar o'z ilmiy yo'nalishining yetakchilari va nufuzli ilmiy maktablar vakillarining ilmiy tadqiqotlari va texnologiyalar maydonidagi chuqur ilmiy munozaralar, olimlarning o'zaro tajriba almashishi hamda tadqiqotchilarning miqyosining kengayishiga zamin yaratadi degan umiddaman. Hurmatli anjuman qatnashchilari, hozirgi murakkab davrda ishonchim komilki, biz bilim va zamonaviy ilmlarni rivojlanishida birlashsak, albatta anchagina kuchayamiz. Ushbu yo'nalishda esa bizning ilmiy hamkorligimiz eng asosiy omil sifatida qabul qilish mumkin. Ilm-fan va texnologiyalarning insoniyat tarixida misli ko'rinmagan darajada rivojlanishida va ilm muammolarining hal qilishda bunday hamkorlikdan katta ahamiyat kasb etadi. Shunday qilib, ushbu xalqaro anjumanning asosiy maqsadi yuqorida zikr etilgan ilmiy yo'nalishlarning zamonaviy muammolariga yechim topish, ilmiy innovatsion rivojlantirish, olimlar, ilmiy maktablar vakillari doirasida aloqalarni mustahkamlash, ilm-fan rivojini yangi bosqichga ko'tarish, Qolaversa, xalqaro hamkorlikni yanada rivojlantirishdan bilan birgalikda tadqiqotchilar, soha mutaxassislari, professor o'qituvchilar o'rtasida o'zaro manfaatli aloqa tizimini yanada yuksaltirishdan iboratdir. Shuni alohida ta'kidlash lozimki, bugungi konferensiyada yetakchi yetuk universitetlar kabi, ya'ni Yaponiyaning Kyushu universiteti, Finlandiyaning Abo universiteti, Yaponiyaning yana Ritsumikan universiteti, Birlashgan Amir Arabliklarining Sharjadagi Amerika universiteti, Pokistonning Azad Jammu va Kashmir universiteti, Amerikaning yana bir Rutgers universiteti, shuningdek ishlab chiqarish ilmiy markazlaridan Intel korporatsiyasining AQSh vakillarining ilmiy ma'ruzalari biz ishtirokchilar e'tiboriga havola etiladi va men umid qilaman va ishonchim komil juda katta qiziqish uyg'otadi. Yana bir quvonarli tomoni shuki, ko'pchilik ma'ruzachilarimiz bu o'zimizning vatandosh olimlarimiz, ularga ham hammamiz nomimizdan katta rahmat deymiz. Hurmatli anjuman ishtirokchilari, ushbu anjumanning o'tkazilishi nafaqat oliy ta'lim muassasalari, balki mamlakatimiz hayotidagi muhim voqea bo'lib, shubhasiz ilmiy tadqiqot ishlarini yangi bosqichga ko'tarishga xizmat qiladi hamda kelgusida yanada ilm-fanni yuksalishiga asos yaratadi degan umiddaman. Barcha anjuman ishtirokchilari va mehmonlarga sihat salomatlik, ilmiy va amaliy faoliyatingizda kattadan katta muvaffaqiyatlar va albatta buni zamini asosi bo'lgan hammalaringizga omad tilayman. E'tiborlaringiz uchun katta rahmat.
Thank you so much, dear Professor Umurzakov, for such a wonderful and inspiring speech. So let me introduce Professor So-Won Lee. Professor So-Won Lee is the current president of the International Union of Materials Research Society. Moreover, now he belongs to the Department of Environmental and Biochemical Engineering at Sanmun University, Republic of Korea. His current research interests include using microstructures, design of Cylon ceramics, of multifunctional properties such as high thermal conductivity and high transparency, the development of photocatalysis by using nanocrystalline oxide ceramic powders, low friction of graphene coating, and electroplating of nickel nanoceramic composites. Please, Professor So-Won Lee.
Do you hear me?
Yes, Professor.
Okay.
Welcome.
Dear Rector Uktan Umurzakov and distinguished professors, congratulations the first conference on problem and perspective of modern science, ICPPMS 2021. I'm so happy to deliver welcome speech today. I had served as President IUMRS, International Union of Material Science Society, in 2017-2018 for 2 years. And then I traveled in the world, such as USA, Europe, Africa, Latin America, Mexico, and Russia, Singapore, all Asians, even Uzbekistan I visited. In order to deliver issue of the materials development and utilization for the next generation. Material research is defined by broad intersection of many disciplines with the material science engineering, including physics, chemistry, biology, mathematics, and other engineering disciplines that usually combine in pursuit of understanding the property of materials and the phenomena they host. Materials are abundant, serving as critical building blocks in technology and innovations. This research impact life and society as it shapes our understanding of the world and enable significant advances in electronics, communications, transportations, and health-related fields. Development and the deployment of advanced materials are major drivers for global economic growth. In light of emergence and spread of coronavirus disease 2019 COVID-19 in the world, there was a huge decline in air passengers. The sky in Europe, the sky in America, sky in Far East, such as Korea, China, Japan, are so clean compared to the previous time. So after vaccination, maybe more air passengers from Europe and America will increase very rapidly near soon. We will use more fuel to produce powers for electric vehicles, even though solar cell and windmills are developing. In battery industry, among China, Japan, and Korea for the electric vehicles, which has been fiercely competing for each other. Competition for the development next generation material has ignited in the earnest. Battery makers are focused on their effort on developing next generation batteries to catch a future market. An official from battery makers said, currently, batteries for electric vehicles have developed a level that can run about 450 kilometers on single charge. So the competition to develop next generation battery with a dramatically improved mileage and safety has begun. We in Korea, we have three major battery companies: Samsung, SDI, SK Innovation, and LG Chemicals. They are working very hard to develop new battery for next generations. So within 200 years after Industrial Revolution in England, we spend too much material such as iron and steel, non-ferrous materials, and fuels and chemicals for develop global economics. But now we should find out how we can reduce the emission of carbon dioxide and decrease amount of materials. So we should think about for next generations. Thank you so much. Have a great, successful conference. Thank you.
Our sincere gratitude to Professor Son Won Lee for such articulate speech you have just now shared. Thank you one more time.
Thank you.
So the next on the line to give a speech is going to be Professor Ibrahim Abdurrahmanov, Minister of Innovative Development of the Republic of Uzbekistan. Please, Professor. Our sincere gratitude to Professor Son Won Lee for such articulate speech you have just now shared. The next on the line to give a speech is going to be Professor Ibrahim Abdurrahmanov, Minister of Innovative Development of the Republic of Uzbekistan. Please, Professor. Our apology, our minister will join to us soon after several minutes. I am pleased to announce that Professor Adil Abdurrahmanov, rector of the Tashkent State Transport University, is next on the line. You are welcome, dear professor.
Добрый день, уважаемые коллеги, дамы и господа. По некоторым уважительным причинам Адул Каландарович сейчас не смог участвовать, но при этом я озвучу за него его слова. Поздравляем уважаемых участников данной международной научно-практической конференции, посвященной решению проблем и перспективам развития современной науки. Считаем, что данная конференция, проводимая при участии Ташкентского института инженеров ирригации и механизации сельского хозяйства, Южнокорейского университета Санмун, Ташкентского государственного транспортного университета и Ташкентского научно-исследовательского института химии и технологий, является значительным событием в научной сфере. Данная конференция затрагивает многие актуальные вопросы, такие как энергетика, гражданская инженерия, агротехника, передовая химическая технология, информационные технологии и другие. Считаем, что главная цель данной конференции заключается в обмене передовым опытом и знаниями в сфере развития инноваций и научной сфере, выявление проблем, стоящих перед учеными, и перспективами развития вузовской науки. Надеемся, что полученные результаты будут полезны всем участникам конференции и предложенные рекомендации действительно найдут свое применение в практической деятельности. От имени руководства Транспортного университета приносим слова благодарности руководству Ирригационного института и всем организаторам данной конференции. Желаем всем участникам конференции плодотворной работы, конструктивного диалога и эффективного взаимодействия. Благодарю вас за внимание. Спасибо.
So thank you very much for your interesting speech. The next on the line to give a speech is going to be Professor Ibrahim Abdurrahmanov, Minister of Innovative Development of the Republic of Uzbekistan. Please, Professor. Please, Professor Ibrahim Abdurrahmanov.
Hello, everybody. Are you hearing me?
Yes, yes.
Sorry, I was on the road, so just entered to university, and I'm having this connection with technical problems. Now it's okay. So first of all, thank you everybody for waiting me for some time, and I really appreciate that you invite me today for this opening speech. Actually, this kind of conferences is very good because innovation, innovative development, science in isolation will be very difficult, and in isolation will not be developed. So today's international conference dedicated to agricultural engineering, chemical technologies, energy technologies, very important for innovative development. Innovative development actually in Uzbekistan picked it up for past 5 years, and our main agenda, main mission, of course, is human capital development. That's why international partnership, this kind of conferences will really fuel this kind of innovations, innovative development of country. For past years, we really worked on straightening up ecosystem for innovation in Uzbekistan. we wrote many documents that really helping for our scientists to do research, first of all, then result of research should be commercialized, and they have all the conditions necessary for this kind of innovative activity. That's why today's conference, I see lots of new developments, lots of good lectures, and I wish all the success for all the participants. And as a Minister of Innovative Development, I am ready to support any conclusions you will make throughout this conference. Thank you very much.
Yeah, we are thankful for such an informative speech, Professor. Thank you very much one more time. So other participants, Professor Azimjon Nazarov, Deputy Minister of the Water Resources of the Republic of Uzbekistan, and Professor Athamjon Khudaykulov, Head of the Department for Development of Research and Innovative Activities at the Ministry of Higher and Secondary Specialized Education of the Republic of Uzbekistan, planned to deliver their speech, but unfortunately they cannot participate in our opening ceremony due to the business trip. They have expressed their excuses. Thank you very much for all of you giving such an informative and wonderful speech.
Да, от Министерства водного хозяйства по поручению министра и замминистра выступит доктор Усманов. С вашего позволения.
Okay, Dr. Usmanov, you may start, please.
Спасибо. Доктор Усманов, начальник управления внедрения научно-инновационных технологий Министерства водного хозяйства Республики Узбекистан. Сами слышали, по уважительной причине и министр, и первый заместитель министра сейчас находятся в командировке, и они попросили меня выступить приветственным словом. Так, поэтому разрешите искренне рад приветствовать сегодня всех участников международной конференции, представляющие институты, университеты и исследовательские организации различные. И особо благодарность за содействие организации столь представительного мероприятия партнерам из Корейского университета Санмун, ирригационного института и других университетов. Безусловно, настоящая конференция является весьма знаменательным событием и развитием науки и образования. Хотелось бы отметить, что с каждым годом интерес к конференциям со стороны нашей студенческой молодежи растет. И это в первую очередь связано с теми широкомасштабными реформами, которые проводятся в нашей республике. Тема сегодняшней конференции является актуальной не только в масштабе республики, но и всего мира. Президент Республики Узбекистан, уважаемый Шавкат Мироманович Мирзиёев, одним из приоритетов во внешней политике Узбекистана обозначил именно решение вопросов совместного использования водных ресурсов региона. За последний год активизировалась регулярная встреча двухсторонних рабочих групп между странами региона по водохозяйственным вопросам. Плоды усилий совместных действий в духе конструктивного диалога и взаимного уважения и интересов соседей проявляются уже сегодня. И за последние годы также подписано более 10 указов постановлений в сфере водного хозяйства. И во всех из них учитывается роль науки, роль образования, роль интеграции науки и производства и водного хозяйства. Так, уважаемые участники конференции, на сегодняшний день наука открывает новые пути и возможности для увеличения вклада в сферу водного хозяйства. Это позволит обеспечить кардинально более эффективное развитие всего общества, что в свою очередь позволит возродить утраченные исторические преимущества отечественного образования и науки, найти адекватные, отвечающие потребностям социально-экономического развития страны, решения возникающих проблем. Особое внимание должно быть уделено интеграции высшего образования и науки, активизации инновационной деятельности, усилению ее воздействия на развитие экономики и социальной сферы. Дорогие участники, искренне убежден, что данная конференция внесет достойный вклад в обеспечение устойчивого развития водных ресурсов региона, послужит эффективной площадкой, призванной укрепить взаимодействие между всеми заинтересованными сторонами, сфокусирует их усилия на сохранении воды как незаменимого ресурса для будущих поколений. Пользуясь предоставленной возможностью, я хочу еще раз поблагодарить всех участников конференции, пожелать плодотворной совместной работы и призвать вас к конструктивному диалогу и активному обсуждению приоритетных вопросов в целях решения существующих и будущих проблем региона, и в частности вопросов, касающихся внедрения инновационных и технологий в водную сферу. Спасибо за внимание.
Thank you very much for your interesting speech, Dr. Usmanov. Now, that's all for the opening ceremony. Thank you one more time, all speakers who took place in the opening ceremony. Thank you very much. So we are start the plenary talks, the second part of our conference. So the 8 plenary talks is planned today. So the next part of the conference is dedicated, as I told, to plenary talks. Please write your questions to me through the chat. So the first on the round is our first speaker, Professor Pankaj Attari. So let me introduce Professor Pankaj Attari. He obtained his PhD in 2013 from the Department of Chemistry, University of Delhi, India. He worked as an assistant professor at the Department of Electrical and Biological Physics in Plasma Bioscience Research Center at Kangwon University in Seoul, Korea in 2016. He won one of the prestigious Marie Skłodowska-Curie Actions Fellowship and worked as a postdoctoral researcher in a research group Plasmat in Antwerp, Belgium, between 2017 and 2019. Now he's an associate professor at the Center of Plasma Nano Interface Engineering at Kyushu University in Japan. Please, Professor Pankaj Attari, you may start your presentation.
Hello, can you hear me?
Yes, Professor.
Okay, so thanks for nice introduction, Jamal, and it's a long time to see you after the Belgium. I can introduce that Jamal and me are the colleagues in the same laboratory in Plasmat in Belgium, where Jamal has done his doctorate degree. So today I'm here to talk about the impact of seed coat color on germination and seedling growth in plasma agriculture. So as Jamal already discussed that I'm working in the Kyushu University and our center is Center of Plasma Nano Interface Engineering. So this is the Japan, and here is the Kyushu Island on which we have the Kyushu University, and this is Fukuoka where the university is, and this is our campus. We have a 4 campus. This is our 1 campus, which is one of the largest and newest campus in the Japan. So the question is, I heard the many invited professors, they deliver that crop yield is a main problem, and the world is dealing with it. So we are currently also working on the plasma agriculture to increase the crop yield. So the question is why it is important. As we know that the land is constant and it's somewhat not, it is decreasing rather than increasing, that more agricultural land is occupied by the industries, where the population is continuously increasing. And since 1980s, the agriculture yield is decreasing. That maybe have many reasons. The few of them are global warming, excessive irrigation, fertilizers, etc. So there is a need to increase the crop yield because the population is increasing. Otherwise, there will be a big hunger problem in the world, and the next war can be happen on the food itself. So that's why it is very important to increase the crop yield to feed the large population. If you put in the scoopers the keywords like plasma and agriculture, you find there is an increase in the publication every year. Suddenly there was a drop due to the COVID-19. That's it's a known effect worldwide. So there's a decrease in the publication in the plasma agriculture field. But otherwise, if we see the trend, it was increasing constantly. So I don't know that how many people are here from the physics part of the nose plasma. So I just give a small introduction that a plasma is the fourth state of matter. And as we know, we have a solid, liquid, and gas. And if further we increase the temperature, it leads to the plasma, there where it has ions, electrons, and neutral particles. So the plasma can be divided mainly into two parts, the thermal plasma and the cold plasma. The main difference is the temperature. The temperature in the thermal plasma is quite high. One of the easiest example is the lightning. So whenever we have a lightning in night, we saw there was the thermal plasma. That's a type of thermal plasma. Even sun is also a thermal plasma. Where the cold plasma is have a temperature between 5,000 and 10,000 between the plasma region, but it has the atmospheric temperature outside the plasma electrode. So as we see that you can even put the finger behind it. The volume of the discharge volume in the thermoplasma is quite high, whereas very low in the cold plasma. Due to the large scale, it is very difficult to handle the thermoplasma, but it's quite easy to handle the cold plasma. And it is literally relatively cheap and safe also as compared to thermoplasma. So there are some basic advantage of the cold plasma that we can use this at the laboratories. So what is the action of the plasma? As I said, that when the plasma generated, generate between the electrons, positrons, and neutral ions. When these react with the air, they generated radicals and UV radiations. And these radicals and UV radiation can attack the biological molecules. So it can be any bacterial molecule, human cells, or any other things. So when these radicals can react with the proteins and enzymes. So when it reacts with the proteins, it leads to the protein denaturation or amino acid oxidation. It can also lead to the lipid peroxidation that can easily destroy the cell. It can also lead to the DNA damages, and it can affect the cell membrane. So through this all different varieties, the plasma can kill the bacterial cell or human cell, or we can say any biological medium. So there is a way to control the reactive species. One of them is the feeding gases. So if we have the different feeding gases, for example, argon, nitrogen, oxygen, or air, we can generate different chemistries. And that chemistry can be vary from the gas phase to the liquid phase. For example, in the argon plasma, if pure argon environment, we have argon plasma, but when it reacts with the water molecules, it generates hydroxyl radical and hydrogen peroxide. On the other hand, in the nitrogen plasma, when it react with water to form hydroxyl radical, and but additionally it can also generate the ammonia. On the other hand, when we have the oxygen plasma, it can produce atomic oxygen, singlet oxygen, ozone, addition with hydrogen peroxide and hydroxyl radicals. But when it comes to the air plasma, the combination is quite high. So we have many more reactive species, as I mentioned here. But some of the major reactive species are the nitric oxide, nitrogen dioxide, nitrite ion, nitrate ion, atomic oxygen, ozone, hydroxyl radical, and hydrogen peroxide. So the mean is that with the change in the feeding gases, we can change the composition of the reactive species, and depend on the composition of reactive species, we can able to control the results. So there is some application that this is an application in the dentistry. When the plasma you can see this example. This is only before plasma treatment. This is surface treatment with H2O2. H2O2 is generally used for whitening of the teeth. It is very common worldwide. But when we use the hydrogen peroxide with plasma, we see very clear, more whiteness is there. So that act as a bleaching, tooth bleaching with the help of plasma. Wound healing is another prospect which is almost commercialized nowadays, where the plasma treatment can increase many interleukins, and it can kill the bacteria on the wounds, and also help in the proliferation of the cells. So after the plasma treatment, there are many markers which are increased, and these markers increased can help in wound healing faster as compared to the normal treatment. So this is an example that the wound healing is quite faster in the presence of the plasma treatment as compared to the control. Then there are applications in the cancer treatment. I just give some example that if we have treated the microsecond pulse plasma activity media to treat the human lung cancer. So this is the HCXO is the lung cancer, and this is the normal. And when we add the plasma treated media, we see there is a change in the morphology. It depends on the concentration of plasma treated media. But there is a morphology is not much change in the control system. And this comes as a cover page of Journal of Physics T on their 50th anniversary. Then plasma has a role in sterilizations. This is an E. coli bacteria, and we have treated with nitrogen plasma with water molecule, nitrogen plasma with nitric acid, parts of the nitric acid molecule to increase more nitric species. And as we see in the same images, the more the structure is completely damaged after the plasma treatment. We have also checked on the multi-drug resistance SRS bacteria. For example, this is the W is wild type, penicillin, methicillin, gentamicin. And in all the cases, we have tested two types of plasma. The NPP is nanosecond pulse plasma and directly buried discharge plasma. And we found that in NPP, then both the cases, the SRS bacteria has died, but more we observed in the NPP plasma. As we see here, the cell wall is completely ruptured. And then we have done the simulations and observed that the bond has changed. So if we see this, the more CO bond is there. So the PG, the peptidoglycan on the bacterial wall is completely modified, is going oxidized. So that is one of the reason why we have more bacterial death in the presence of the plasma. Then we also checked on the thermophilic bacteria proteins. Actually, those who don't know, thermophilic bacteria, those bacteria that do not died with the high temperature, but they can easily survive in high temperature. So that is very hard to sterilize because in a general sterilization method, we generally heat the system, but the thermophilic bacteria still survive. So to degrade that, we use the plasma and we check the proteins of the thermophilic bacteria modification. And we found that the histidine 52 and tyrosine 59 of the molecules are completely modified after the plasma treatment. And this is the last year results from the US group where they used the cold plasma on SARS-CoV-2 inactivation. And they have treated the plasma on the different surfaces like plastic, metal, cardboard, football, baseball, basketball. And with the pace of maximum up to 180 seconds, all bacteria die. So SARS-CoV-2 is killed by the plasma. So the question of the main of this today presentation is the plasma agriculture. So the motivations we have motivated why? Because it is firstly is the low cost and increase the production rate. So the energy consumption is only about 90 joule per gram, which is approximately about 0.14% of energy required in cultivation in harvesting process. This is an example in our laboratory. We have treated the plumia seeds after the plasma, and after 2.5 years, there is a big difference between the plasma and the control. And we have also checked this rice, and we found that after the plasma treatment, 49 days, there is a quite difference in the in the rice plants, and it observed that the 70% they are increased above the ground level after the plasma treatment. So these are very interesting results which motivate us. This is another example of a laboratory where we found we had treated the arbutosis seeds with the plasma, and we found that 11% harvest time is reduced and 56% crop yield is enhanced. So that's very good results we obtained, that after the plasma treatment, the yield is also increased. Not only the harvest time is decreasing, but yield is increasing. And I think that is one of the most important criteria for the plasma agriculture. So today I will talk about the seed coat play, how seed coat plays and filter of the rones, or the transparency of the seeds. So many times it's observed that when we treat the different seeds with the plasma, we observe different results. So we try to analyze that what is the effect of seed coat color. Is seed coat color can affect differently, or there is some other also physical parameters that affects the plasma effect on the germination and growth. So this work is recently published where we studied the impact of seed coat color and the storage time. So we use the different storage time like 2017 harvest and 2018 harvest, and we done this test in 2020. So and we also differentiate between the seed coat colors. So this is our experimental condition. We took the radish seeds and divide into two colors, brown color and a gray color. We use our DBD, and the seeds are not put directly down the DBD. They are 3 millimeter away the DBD. So that means that there is no direct effect of the heating effect or the electric effect. Only we thought that this is the Ron's effect has mainly plays the most important role in this type of in the germination of radish sprouts after plasma treatment. And this is the physical parameter where the power density is about 3.0 and the radiation time is 180 seconds. So this is the two type of treatments, the harvest year of 2017 and 2018. And we found that in 2017, there is an 8% increase in a gray seed after the plasma treatment in germination. Whereas in the 2018 harvest, the final germination after 72 hours is same, but after the 20 hours, the brown seeds, we observed the 50% germination, where the gray seed we observed at after 32 hours. So the main thing is that the germination is almost the same in the 2018 harvest, but it is different in 2017 harvest. So that shows that between the brown and gray color seeds. So both are radish seeds, but they are divided in brown and gray, and they have the different rate of germinations. Later, we measured the mean and shoot lengths. We observed that in the 2017, there is a 50% difference between the brown and a gray seed in a root length. And after the plasma treatment, there's 13% increase in a brown and 25% increase in a gray. But in the 2018, there is no significant difference in the brown seeds, but the gray is decreased by 16%. For 2017 harvest also, we have observed the 59% difference in a controlled brown and controlled gray. And after the plasma treatment, there's 59% increase in the gray seeds of the plasma treatment. So this shows that the mean root is increased by 25% and shoot is increased by 59% in the gray seeds. So our study saw that in 2017 means those are older seeds. It has more important action as compared to the younger seeds. Later, we have studied the EPR. The EPR to see that is the seed coat has some differences. So there is no new peak is generated in plasma treatment. And we observed the two peaks like at G4.3 and the G2, and this is MN peak. There we found there is some change in intensity. So at G2 in a gray seed, the increase by 36%, and for the 2018, the increase by 32%. This is a semiquinoline. This is a semiquinoline product. So during the plasma treatment, the more semiquinoline product has formed, that results in an increase in the intensity. Further, we have checked the Fe3, that is at 4.35. That is mainly the increase in Fe3 in the gray seed larger than brown seed. So there is a 97%, 94%. And this is the chemistry where we observed that the F is mainly in the ferric pyrite form, and during the plasma treatment, the more radical formation has occurred. That can result in an increase in intensity after the plasma treatment. Later, we have checked the phytohormones and gamma-torficol. The gamma-torficol is a type of vitamin A. It is we can say antioxidant. And the amount of GA by ABA is a main ratio for the germination. So if, as we see previously, in 2017, there is more germination in the gray seeds. So when we check the GA3 by ABA concentration, we observed there is a 416% increase. Whereas in the 2018, in the brown seeds, where we observed the increase in shoot length, there is an increase of 232%. Whereas in both the cases, the gamma-torficol decreases. It means the antioxidant is decreased, whereas the GA3 by ABA has increased after the plasma treatment. Later, we have checked the SAM analysis, and we didn't find any much change in the SAM analysis except the gray plasma. The surface is quite ruptured, but in the rest of the cases, we didn't observe any significant changes. So what is the mechanism? If GA by ABA decreases, it leads to ABA pathway. That means seed dominancy. The germination will delay. But if GA by ABA increases, that leads to germination. So in our plasma treatment, the increase in the gray seed 2017 and early germination in the brown seed in 2018 is because the GA by ABA ratio has increased after the plasma treatment. And this is the short data I will told that about the delayed seed after the stress in this of the rice seed. So the heating effect. So when we have taken the productive and we sow it and then the vegetative growth, we got the seeds. But we observed that during the seeds of the heat treatment, or we can say the hot temperature, the germination is decreased in the rice seeds. So as we know, the temperature, climate changing is increasing. So we observed that in Japan also, the climate change will increase quite drastically. And that can result in a reduce in the rice field yields because of the heating effects. So what we thought, we treated this seeds with the plasma and heat and check how it behaves. So this is the germination, this is the control seed, and this is the heated, and this is heat plus plasma for 3 minutes. And we observed that the increase in the germination we observed after the plasma treatment. After the 72 hours, then as I already discussed, generally there is two main things for germination is AB and GA. So we checked these genes NCD and ABA-OH for. So if NCD increases, that leads to the ABA. If decreases, that leads to the then if this dominant gene is scanned, then ABA decreases. Similarly, GA20 and GA3 can lead to the GA enhancement. And these are all AB and GA related things. And then alpha amylase that also help in the germination window. So to know that, okay, these factors are affecting after plasma or not. So we have taken seed germination and done the gene expression analysis. And we observed that after plasma plus heat, the NCD is decreases, which can lead to the ABA. So we can say that, okay, fine, because ABA this gene expression decreases, means ABA is decreasing. Then we taken another, so here there is no significant difference. So if this is increase or decrease can affect ABA, but it does not have any change, then we taken the GA genes and we observed that there is no significant changes. Another GA genes we taken, there is also no significant changes, even though there are slightly decreases. And in the alpha amylase gene, also we didn't find any significant changes. So that means that plasma effect on radiation is something different, which we are expecting. So the plasma radiation has less effect on gene on GA biosynthesis. It means that there is a high germination, but it is not through GA. So there is some other factors, might be some epidemic changes are there. So to understand this, we done the methylation test. So we treated with the plasma and we found the DNA methylation level decreases. That can relate to the germination. So there it decreases after the plasma treatment. So here my summary. We have the seeds. The plasma radiation induces hypomethylation that improves the germination, and the sensitivity of the plasma can depend on the seed coat color or the pigment there. So this is the case that during the heat stress, the DNA can methylation, but after the plasma treatment, there is a hypomethylation that can increase the germination. Thank you for attention, and I will thankful for ICPPMS organizing committee and Jamal to give me a chance to present my work. Thank you. If you have any question, I'm happy to answer.
Thank you very much, Professor Pankaj, for such interesting talk. Apparently called atmospheric plasma has been using in various applications. So as you know, we are planning establish a collaboration with you. Hope you will help in this issue in the future.
Sure.
So questions, please. I don't see any questions in a chat room. Okay, if there is no questions, then Pankaj, is the rice mainly seeds that you are treating nowadays, or are you testing another seeds as well?
Actually, we are doing different tests, but the rice one we are doing from last 4 years. So this is like 3rd generation and 4th generation we are also treating. So we are doing actually land experiment data. I didn't show that data yet. So we are doing actual land experiment data that how the 3rd generation or 4th generation treatment can affect the germination rate and its tendency to adopt the environment under the different stresses. So this is one of the thing. And for the laboratory level, we use tomatoes, radish, arbitoses for the standard tests. Yeah, so 3-4 seeds we are generally focused right now.
Okay, thank you. And I also faced one problem. I also applied this kind of plasma agriculture project in my country. So there is a interesting question arises. So plasma treatment increases the seed surfaces hydrophilicity, right?
Yeah, one of the reasons.
So if it increases hydrophilicity, it adversely might affect to the seed, because if the raining season, for example, come too frequently, then after excessively wetting of the surface might occur. In this case, seed might die.
Actually, yeah, I understand your point, but the seed surface, the hydrophobicity is increasing, but all seeds, they have some limitation. They don't absorb water from more than one level. So if you put your seeds for 1 hour or 5 hour or 6 hour, after 1 hour you don't find, depend on the seeds, but you don't find much differences. So the after treatment, if you see that seed can absorb very excess amount of water, that is not there actually. And moreover, we treat the seeds before the sowing. So we not treating it and keep it very long time. We do that test also actually to keep in a long time at -80 degrees Celsius. And when the time comes, then we used to sow it. But generally, it doesn't affect much. It has some limitations. More than that, it never absorbs water.
Thank you for your answer, Professor, and giving such interesting talk. Is actually one of the innovative way to increase crop on the field by applying plasma. And thank you very much.
Thank you so much.
See you soon. Maybe this year or next year.
Yeah, sure.
Thank you very much.
Thank you. Well.
Right. So our next speaker is Dr. Parthiban Marimutu. Dr. Parthiban has completed his graduate studies focused on structural investigation of human neuronal alpha-7 nicotinic acetylcholine receptor in response to neurotoxins and neurotransmitters. After completion of his PhD, Dr. Parthiban took a short-time research visit to McGill University, Canada in 2009 as an exchange researcher, and the Max Planck Institute for Molecular Genetics, Germany in 2010 as a visiting scientist, where Dr. Parthiban had a chance to lead a group of researchers focusing structural modeling for Cas9 competition. So next, Dr. Parthiban has started his postdoc career during 2011 at Abo Academy University in Finland, where he spent significant period of time focusing various aspects of structural bioinformatics, in particular MD simulations and X-ray crystallography studies as well. Next, Dr. Parthiban has moved to Albany State University, Albany, Georgia, in USA in 2019 for 1 year for his second postdoc period, where he investigated the conjugate series of organophosphate compound for its structure, activity, relationship, and toxicity. Further, Dr. Parthiban has returned to Abo Academy University, continued his postdoc starting 2018. Last summer, Dr. Parthiban obtained the title of docentship in the subject structural biology and computational biochemistry, and continue to focus on various aspects of MD simulations and supervising several graduate, masters, and exchange students for their thesis work. All his research involves collaboration with national and international researchers. We also started our active collaboration with Dr. Parthiban in 2019. Please, Dr. Parthiban, you may start your presentation.
Okay, I'm just sharing my slide. Can you please confirm my audio is clear and my slide is visible?
Yes, Parthiban, everything is perfect.
Okay, yes, yes. Thank you very much. I would like to turn off my video during my presentation, and we can have our questions once the presentation is over. So good afternoon to everyone, and I thank you very much for inviting me to the web lecture series on First International Conference on Problems and Perspectives of Modern Science. I would like to thank the organizers for providing me the opportunity to present some of my research highlights to the community. Okay. I am Dr. Parthiban Marimutu. I am a docent at Abo Academy University, Finland. Today, I'm going to talk to you about an introduction to the computer-aided pharmaceutical research. In a bird's eye view on leading computational chemistry tools, application of leading computational chemistry tools to pharmaceutical research. If you look into the Finland's map, which is on the right, on the lower left corner, where we are, which is the southwestern part of the country, and here is the external appearance of our science park where I conduct my research. Moving on to today's lecture topic, and at the end of the lecture, I believe you may all able to gain a little bit of understanding towards the need for in silico approaches in drug design and different in silico methods involved in the drug design, and how to make use of the existing chemical data which is available in the internet for the drug design, and how to screen the chemical databases and understand the behavior of drug with its target protein. It will be appropriate to describe the different experimental research methods used in today's drug discovery approaches with the classic blind man and the elephant parable. I mean that each methodology often isolates different properties that fails to give a complete picture. That is, one blind man touches the tusk and recognizes it as a snake, while the other touches its ear and recognizes it as a rug, and so on. They don't actually meet and make a complete picture, in my opinion. While the computational methods can bridge the gaps between the different experiments and bring it to a complete picture. In today's scenario, several people are suffering from several diseases that may have emerged from different origin. Example, allergies, autoimmune disease, and cardiac disease, cancer, and based on metabolic deficiencies, and etc. And sometimes the drugs that are provided for the treatment of these diseases gain resistance, which is worse. This increase in the number of diseases day to day that leads to even corona today, which is going on right now, ultimately keeps highlighting the fact that there is an urgent need for novel drugs consistently. The conventional approach to drug design have several stages, and it is postulated that this conventional approach will need at least a period of 16 years to produce drug and bind it to and bring it to the market. Today, computers are used everywhere, as everyone knows. Similarly, computational resources are employed in the field of drug discovery as well. The computers uses advanced and sophisticated algorithms that are significantly fast and cost effective, that can reduce its huge time period. Today, computer methods that I planned to talk to you all have shown a remarkable decline in the time period used by several conventional, that is, experimental approach. Getting a little bit into the subject, among the various diseases that we have seen previously, I work in cancer, and I'm planning to use that as an example. And cancer, also called as apoptosis, that are prevalent disease worldwide, that constantly being a massive threat to the mankind over a period of time. Major reason behind is the resistance to the chemotherapeutic agents. The literature says that the apoptotic process occurs in human body via two different pathways: extrinsic and intrinsic pathway, which takes place by two different mechanisms and subsequent cascade mechanisms. The BCL2 family proteins, in which I am highly focused on, are the central regulators of apoptosis, in which falls into the intrinsic pathway. The BCL2 family proteins are subdivided into three groups, as can be seen here: anti-apoptotic protein, pro-apoptotic protein, and BH3 only proteins. This classification is mainly based on the presence of a number of BH3 domains. Structurally, the BCL2 family proteins are highly similar, which means that the family of proteins are tightly packed alpha helices, helical bundles, among which a certain group of residues forming the alpha helices 2, 3, 4, 5, 6, and 8 forms a hydrophobic binding groove on the surface of the protein. This binding groove involves the binding partner selectivity, in this case engaging a BH3 domain of pro-apoptotic proteins in response to the signal. Although all pro-apoptotic proteins form amphipathic alpha helices, their amino acid sequences varies to different degree. In a normal cell, the BCL2 family proteins are mainly localized in the mitochondria, and when the cell experiences stress or based on signals such as a targeted therapy, chemotherapy, oncogenic inhibitions and stress, etc., the binding of anti-apoptotic proteins and the pro-apoptotic proteins takes place. The binding causes the release of cytochrome C from the mitochondria through mitochondrial outer membrane protein that further leads to apoptosis and eventual cell death. The binding of pro-apoptotic protein and anti-apoptotic protein is crucial to decide the fate of a cell. This means that there must be a proper balance between the binding of binding of pro-apoptotic protein and anti-apoptotic proteins. The imbalance in this binding is the initiation factor to the cancer. That is, it leads to the overexpression of the normal or pathological cell survival that leads to tumor formation in the different parts of the body. Therefore, these family proteins have the central role in various leukemic conditions. It is believed that during the drug discovery process, while specifically targeting this protein binding pocket, there is a high chance to suppress the tumor formation. Now we are stepping into the methods part of the lecture. Here is the schematic representation of different methods used in the computational drug discovery process. This method primarily depends on the information available regarding the disease-related target molecule, that is based on the literature. One of the method is the ligand-based drug discovery approach, which uses existing chemical data information to start with. This method comprises 2D QSAR, 3D QSAR, pharmacophore mapping, fragment-based and scaffold hopping approaches. And the next important method is structure-based drug discovery method, which needs structure information for of the disease-related target protein. And this method comprises structure prediction, binding site prediction, molecular docking, high-throughput virtual screening, and molecular dynamics simulations. Once the hit compound is obtained after using either one of these methods, the hit compound can be used as a lead generation and the PK/PD testing, and which means pharmacokinetics and pharmacodynamics, which represents pharmacokinetics represents how the body responds to the drug, while pharmacodynamics represents how the drug responds to the body. And further, the ADMET studies that shows how effectively the administered drugs get absorbed, distributed, metabolized, and excreted. The quality test for the drug based on Lipinski rule of 5, which is listed here. This process is very helpful in filtering the drugs that have toxic side effects. And in comparison with the conventional approach, the researchers don't have a straight away to test the drug with the model animals. Once the filtered compound clears all the laboratory testings and clinical trials, the drug candidates can be used for further testing process. Let me describe a little bit more about the ligand-based drug discovery approach. Among these techniques listed here, the 3D QSAR and the pharmacophore model are the commonly used method for identifying potential drug compounds. The steps involve data collection, scaffold generation, and the 3D QSAR model generation, predicting the activity and hit compound generation. On the other hand, the pharmacophore modeling comprises pharmacophore model generation and screening the chemical database against the pharmacophore model and the hit compound identification. Let's get to know more about the data collection procedure. The advantage here is the existing chemical data information used for the target of interest are available online. So the chemical data can be obtained from the database for the target protein of interest that should have a wide range of binding affinity value and a common scaffold. Here I will use one of my recent published results as an example to explain this. So MCL1 is an anti-apoptotic protein that we have previously looked into that is involved in cancer regulation. For that, Freiberg et al. Has synthesized and published 66 compounds that satisfy the above criteria. The highly active compound here have 0.005 micromolar, while the least active compound have 214 micromolar activity. The SMILE codes of all the compounds were used for generating the model. Next, all the 2D SMILE compounds were converted to the low energy three-dimensional conformational pose. The structure, several conformers were generated for each compounds. ConfGen program from the commercially available drug discovery suite Schrödinger was used for this purpose. The generated conformers were further used to develop a common pharmacophore hypothesis that are identified as essential features responsible for the activity. Usually, a hypothesis can be built based on the pharmacophoric features as a acceptor, donor, hydrophobic, negative, and aromatic, and the user can also customize these features. The pharmacophore was separated certain distances and angles in three-dimensional space. The reliability of the pharmacophore model can be tested by superposing the compounds on the hypothesis. This method shows that all the actives mapped nicely, while the inactives don't. Also, it loses certain features. This signifies that when this model was used for screening purposes, it can produce more active hits over the inactive ones. Usually, the suite generates more pharmacophore models, and the user can select the suitable model based on the scores. Now, the developed pharmacophore model can be used as a query and used one or combination of the chemical databases to identify chemical hit compounds that have similar pharmacophoric features. The program have several default parameters that can be used for screening the chemical compounds. Both, but most likely the default parameters will give the potential hits. I would like to point out that it is most difficult to obtain a lead compound this quick by conventional approaches. For this, a high-powered computational resources were used. The next method is 3D QSAR. This method works based on the principle that the structurally similar molecules tend to show similar activity. The advanced statistical approach is used to predict the activity values of these compounds that can be used further to predict the activity of the new drug analogs that can save a lot of time and money. At first, as a first step, the dataset can be split into test and the training group, and the training dataset is used to build a 3D QSAR model, and the test set is used to validate the model's accuracy. It is important to verify how the predicted values of the training and the test set correlates with the experimental value. The correlation values here were high for both the cases. That shows the reliability of the model's predictive power towards the new hit compounds. Now we have a hit compound retrieved from the public database using the pharmacophore-based screening process. It can be taken further to PKPD analysis. Now we have seen. Now we can see how to validate the identified hit compound using the methods from the other approach. One of the classic and most popular technique is called homology modeling. And if we ask ourselves what is homology modeling, in short, we can gain the understanding is constructing a protein model from a known 3D protein structure of homolog protein. When to perform a homology modeling? So when there is no structure available for a protein of interest, and only a small fragment of a protein is available, and only a domain of a protein is available, and only some part of the proteins are missing, we can use the homology modeling approach to construct a protein. Here is the critical steps that is provided, that is listed here or shown here for the constructing the homology modeling approach. Over here, we have the target sequence, and we need a template structure, and we need to align the target and the template sequences that can be provided to the homology modeling tool. It can be, there are several tools are available. and we can generate a target. We can generate a target model, and the best model can be selected based on several scoring parameters. Here is one example of building a full-length model of a protein structure when only a part of it were experimentally determined. Integrin is a multi-domain protein that have high biological significance, but there is no full structure available till date. But using homology modeling approaches, I have constructed a full-length integrin alpha V beta 3 subunit for the first time, and it can be used for further studies. In the second example, I would like to show that this is the MCL1 protein structure, and in one of the structure, it is determined with no ligand, and in the second structure, it is determined with a ligand, but there is a missing region. But we are interested in having a full-length protein with the drug compound that is bound to it. In this case, the homology modeling can be used effectively. Using homology modeling approach, the full-length MCL1 protein model is obtained, where the part of it were constructed using two different proteins. In this way, we can leverage the homology modeling technique to obtain low-energy protein model. The next powerful method is docking. So, what is docking? It is an in silico method used to predict a low energy conformational pose of a chemical compound with its target protein. And when do we perform this docking calculation? When there is a new hit compound identified by other approaches, and when there is when we don't know how its low energy binding pose conformational pose is, and the potential information is used to improve the performance of the identified hit compound. And what are the types of docking available prevalently? The major methods are protein-protein docking, protein ligand docking, and protein DNA docking. And here I have listed few docking programs that are respectively used for the methods provided here. And this is one example to show how a drug compound is searching its low energy conformational binding pose. Oops. For some reason, it is not working. Okay, let's move further. The docking result can be analyzed based on a cluster-based approach to identify the hit compound. For this, the advanced statistical method, the principal component analysis, can be used. The docking is a robust technique. That is, it mainly used to predict the low energy conformational pose of unknown ligand in the binding site of the protein. The anti-apoptotic MCL1 complex with a similar chemical compound was used as a structural framework, and a co-complex ligand is used as a grid center. So the grid, which means it's a virtual box targeted or centered by the user that permits the ligand of interest to search its low energy conformational pose. This can be performed into two different approaches, selecting the surrounding residues, or in this case, of co-crystallized complex. The co-crystallized chemical compound can be used as a grid center, which can also be called as guided docking. In this case, the co-crystallized compound was used as a grid center to perform docking runs. Then the docking results can be compared with the X-ray pose that shows that the hit compound identified by the pharmacophore model screening and the co-crystallized compound occupies similar conformational pose. In this way, we can leverage the docking tool as a method to obtain low energy conformational pose for a newly identified compound. The drug binds nicely inside the MCL1 binding pocket. Also, the drug exhibits electrostatic interactions and the pi-pi interactions with the surrounding residues. We can verify the stability of all these interactions via other advanced techniques and a powerful technique called as molecular dynamics simulation. So what is molecular dynamics simulation? Means like it is a computational simulation technique mostly used or on a wide range of biomolecular system that works based on Newton's equation of motion. And when to perform this molecular dynamics simulation? So when we are the technique is mainly used to understand the behavior of the molecules over a period of time by applying velocity on and forces on each atom of the system in the system. The functionally distinct receptor conformations are not available for most of the receptors of a therapeutic interest. In that case, like we can use this technique effectively. The MD simulations can be designed when one have a 3D protein structure simulation program available and high computational facility and the visualization tool to track the simulation. The steps involved for MD simulation is assigning a force field for a protein and a drug. So the word force field is a new word. So let's see what is a force field means. Despite the fact that the proteins involved in the diverse biological function, all proteins are built with a common building blocks called amino acids. Every atom in the protein is connected by number of bonds and are separated by certain angles, dihedral angles, and they are integrated by electrostatic forces and van der Waals forces. All these entities are constructed in the form of universal computational algorithms that can be applied to any protein for simulation purpose effortlessly. It can be called as a force field. Once we apply the force field, the protein and the small molecule should be placed inside a three-dimensional virtual box, and the solvent should be added. On top of it, we need to add the counter ions to neutralize the system. We call this complete setup as a system. This whole system should be subjected to energy minimization to obtain the system without any clashes or bumps. Once we perform the energy minimization, we subject it to equilibration by applying constant number of pressure and the temperature. Finally, we perform an unrestrained production run for a long period of time, and the results obtained from the MD simulation can be the results obtained from the MD simulation needs to be analyzed. For some reason, the simulation movies are not working here. I apologize for that. I cannot able to figure out why now. Once the MD simulation is over, the simulations needs to be analyzed, and there are several basic methods that can be used to analyze. So they are called root mean square deviation and fluctuation. I'm not going into those things, but the results can be related back to the results can be related back to the experimental results. Here, by comparing the initial and the final pose of the hit compounds, that shows the interactions that are maintained well inside the binding pocket. And the most importantly, the MD simulations provide the opportunity to estimate the binding free energy for the protein and drug compound within the binding pocket. This can also show the crucial residues that are contributing to the binding. This information is very important. This information is very important at the time of drug discovery in terms of substitution. A substitution is crucial for the drug compound. And do we still have time, Jamal? How much time we still have?
You may continue, participant. You have time.
Okay. Yeah, okay, sure. Thank you.
5 more minutes.
Okay, sure. Thank you. what I'm showing here is one of the examples that we have published in collaboration with Zamonaviy. So in this study, this research group has Bonsu research group has performed experimental study of all anti-apoptotic protein against the pro-apoptotic proteins, and they have estimated the binding capacity of these protein peptide complexes. They have studied this in this whole experiment with in the mouse system. So we are interested to know what are the hotspot residues. So we have used molecular dynamics. We have used MD simulations to estimate the hotspot residues. So the energy values for the binding site residues were plotted for all the complexes. It is interesting to see that the two hydrophobic residues lining the binding groove of the protein from both the sides exhibits higher binding energies in all the complexes. Also, the arginine residues contribute to the higher energy in all the complex. And the reason is the interaction between the protein and the peptide was tightly maintained by the salt bridge interaction. And this is another research work we have, me and Zamonaviy have collaborated and carried out. I would like to show this results as well. In this studies, the experimental group has performed binding of pro-apoptotic proteins and the binding of a pro-apoptotic protein with the BIM peptide with wild variety, and they have tested 7 different types of mutants. So we are interested to know like how the mechanistic basis of these 7 different peptide affects with the help of simulations. So what we have done is we have applied steeped molecular dynamics simulation that shows the stepwise release of the peptide bound from the MCL1 binding pocket here. So over the course of simulation, the peptide slowly started to dissociate from the protein binding pocket, and we have investigated its force required for the displacement. And in addition, we have also investigated its interactions closely. So finally, we have compared the results of all the peptides, but in this case, like we have displayed only the results of wild type with the double mutant, which is a double mutant, which is a cyan, and the other mutant which is leucine to the tyrosine, which is in the orange color. So what we have observed from this one is like we have seen that the wild variety is having a higher binding affinity in comparison with the 2 different mutants that we have studied. All my works produced these publications in last 3 years, and few are yet to come. So I would like to stop my my presentation here, and I'm happy to take further questions. And I would like to conclude my talk with mentioning that the advanced computational programs, application of advanced computational programs can reduce the time and the cost in the drug discovery process. And having the privilege to replicate the experiments several times, and the computational methods has the computational methods can be used to test several drugs in a short period of time, and also can study the behavior of the drugs on several targets simultaneously. And all the process or requires high performance computations with less cost effective method that can be achieved. I would like to acknowledge my supervisors, my mentors, and my collaborators, and my computational facility, CSE, for my computational facility for the grants they have provided, for which I've used for all the MD simulations. And I'm happy to take any questions if you have any.
Thank you very much for your interesting talk, Dr. Parsivan. Nowadays, drug discovery problem is being quite modern in the field. So I don't see any questions on chat, but let me ask one question. So
yes, please.
As you know, artificial intelligence applied on protein folding processes,
yes.
And scientists achieved a breakthrough by predicting almost similar 3D protein folding process.
Yes.
Are you also planning to apply artificial intelligence in drug discovery? It's how it's far from applying artificial intelligence in drug discovery, or do you know similar investigations, application of artificial intelligence in drug discovery field?
Okay, well, it is a fantastic question. To keep the drug discovery process in the limelight, it is good to bring the artificial intelligence approaches in the drug discovery process, of course. So to answer the first question, like of course, yes, indeed, I am planning to incorporate the AI methods and the deep learning methods in my future drug discovery process and approaches. And from my side, like me being a biologist, like I definitely need to take a little bit more time to explore the AI methods in incorporating in my research in future. But yes, there are some ideas to consider the AI in my future research.
Thank you for your answer. Hope it will be available soon in drug discovery field. So
yes,
if there is no question, we will continue our conference. Thank you very much for your interesting talk, Dr. Parsivan, and we will maybe meet soon in Europe.
Yes,
this or next year.
Thank you very much for the opportunity provided, and I'm glad, and yes, thank you.
Thank you. So our next speaker is Dr. Abdurrashid Mavlanov. He obtained his PhD at University of Leipzig in 2012 and continues his activity as a senior researcher at Physics Technical Institute in Academy of Science of Uzbekistan. Further, he won a fellowship and worked as a research associate at University of Electronic Science and Technology of China between 2018 and 2020. Now, he is a senior researcher, Ritsumeikan University in Japan. Title of his talk is "The Future of Solar Energy as Bright as Sun." Please, Dr. Mavlanov.
Thank you very much for kind introduction. Assalamu alaikum, dear conference organizing committee and participants. So can you see my slide?
Yes, it's okay.
Yeah.
Yeah.
So as shown in the title, so we are working on solar cells to increase its efficiency and functionality to make it available for everyone. So now we are working in Japan at Ritsumeikan University. So it has several campuses. We are in Biwako Kusatsu campus. And here is the top view of our campus and our lab located here. And solar cells or photovoltaic cells like is directly converts the sunlight into electricity. So we have several ongoing research projects. Today, just I want to briefly introduce one of our project, high efficiency CIGS solar cells. And before that, I would like to just compare the comparison of energy sources, then come to our recent research projects. Here, if we see the traditional and new energy sources, and we can, I think, first questions are what are the safest and cleanest sources of energy? Like traditional energy sources, coal, oil, natural gas, and biogas, it seems they have high rate of accidents, which even can cause death. Or on the other hand, they also have the air pollution. Like for long-term use, we all aware that the greenhouse effect caused by the excessive emission of carbon dioxide. And on the other hand, the renewable energy sources, solar, wind, nuclear energy, or hydropower, they have low accident like reports. For nuclear power, this number also includes accidents in Chernobyl and also Fukushima. And like they have a very good report. But the question is how their next question will be their price. What is the price, and are these new sources can be competitive with traditional resources of energy? Here we can see the price of electricity from new power plants. So it is here expressed with levelized cost of energy. So this means if one person has like a power plant or wants to build, every power plant has a lifetime. The lifetime and also the building of the power plant itself and its running cost, like fuel, everything should be counted, and the producing electricity will have the minimum level of cost. So below this, it will not be effective. Like we can see, the coal is the biggest electricity source in the world, like 33% of electricity generated by burning coal. From 2009 to 2019, within 10 years, its price just decreased for 2%. And this decrease mainly to reduce the cost of coal transportation. But within this time, the coal price steadily, slowly increasing. As a result, there is almost a very small change. And on the other hand, the the second biggest electricity generation power plants, their price reduced significantly. For the combined cycle, or like it is the biggest share for 32%. This decrease is mainly due to the two factors. The first one, the reduced price of gas, and second reason was increased amount of supply. But even the lowering cost of today's data, one can also see in this reference that the gas price is nowadays price is 3 times higher than 30 years ago, if you compare. On the other hand, the nuclear power plants price is increasing. This is mainly due to the new regulations. Now, in many areas of the world, it's very expensive or difficult to build new power plant. And if we just compare by adding solar energy and wind, like 10 years ago, it was very high for solar energy, like almost 360 US dollar per megawatt hour. And for wind power plants, it was also higher than coal. Now, in this day, it is both of them reduced like significantly. For the solar, it is like 89% declining compared to the previous price. And now both of them is very competitive with the traditional power plants. And one can ask, what is the reason for significant reduction? There are like two main reasons. Like recently, the number of production or amount of production is increasing. As we know, for all production products, so with increasing the numbers, so the number of products, the price will decrease. And another thing is continuously increasing efficiency. And on the other hand, there is one more reason, like for traditional coal and gas power plants, if one builds such a power plant, their source, like every day the coal should be burned. So one should get the coal or gas from inside the ground. But in the case of solar and wind, the sunlight and wind comes to the power plant and it's free. But for the coal, for example, for coal-based power plant, 40% of expenses goes to the just buying the coal. And here we can see the clear picture how the price of solar models is decreasing from 1976 until this date. This is mainly with increasing the production lines. Until recently, it was very high compared to traditional electricity generation systems. But as shown in previous slide, it is it is now available. So here is another example. Every year, new renewable energy capacity is adding in the worldwide. Like in last 2 years, we can see the reduced price of wind and solar power plants affected also the new capacity lines. So in last year, the biggest share was for wind and solar power plants, which is significantly higher compared to hydrogen or other combined renewable energy sources. And actually, so solar cells, these solar cells in a few centimeter square area, these are developed in laboratories, or like another term is model. Like if we in the manufacturing put the high efficiency solar cells together to make model, and or putting the group of models making model, or even bigger groups making the solar arrays. So now I slowly zoom in to our research topic. So we are in the lab working on solar cells. Like the motivation for our current research is nowadays we all know that normal solar cells we call it heavy, like for square meter its weight is more than 10 kilogram, and it is rigid. Usually rigid means we cannot bend, or it's not flexible like this. Only for flat surfaces we can use nowadays. But our motivation, how to make it lightweight, like let us say 1 kilogram per meter square, and make it flexible. So it will be it will increase the applications for roofs with low withstand, like load, such as factory roofs. And if it is flexible, it can also be applied for wall surfaces, and also it can be applied for automobile applications. So for this purpose, we choose kappa indium selenide based thin film solar cells. Actually, in the research laboratories, there are many solar cells. Like only here, the successful or high efficiency solar cells are mentioned, like gallium arsenide based or silicon based, or the thin film technologies, emerging technologies like perovskite, and the research laboratories who report these record efficiencies also given here. And why we choose CIS or CIJ solar cell? So it has a one of the key players like silicon, cadmium telluride, and CIGS now not only have the high efficiency, they also available in the market. And CIGS is one of them, and it has continuously increasing efficiency. It is less toxic compared to cadmium telluride. Only selenium is has small toxicity, and also it has very good stability, which is important for long-term production. And in normal like solar panels, only around 3 micrometer thickness in the middle of solar panel is actually the working part of the solar cell. Below and on top it, so there encapsulant and also front glass and substrate glass. These are the main heavy parts. So in new approach, we just developing how to make it thin, the supporting parts, and also make it flexible. However, in this standard structure, like usually solar cells fabricated as a sandwich-like, several layers grown on top of each other, and the high efficiency, about 20%, is shown above. They achieved over the more than 40 years of research achievements. Just simply changing the order of these preparation steps will significantly reduce efficiency. Instead of just changing the solar cell structure, our approach is to use a lift-off process. For this, we don't change the standard fabrication process. We just first fabricate the solar cell with optimized growth conditions, and to put transparent glue such as epoxy, and also put on top of glue, put the fluorine-based polymer films. They are flexible, and then with mechanical stress, we can just peel off the solar cell from its glass substrate. Here is the example of this. Like first, standard solar cell is fabricated, then polymer film is attached on top. Then the solar cell is removed from glass. This is the lift-off process. And then now the lift-off part of the solar cell has no back contact. For this solar cell, if we use another transparent electrode back contact, so it can be not only flexible and lightweight, but it will be bifacial. Like we can keep the high efficiency, we can make it lightweight, flexible like this, we can bend it, and also it will be bifacial. From both sides the light can enter, and we can generate solar energy, the electricity from sunlight. So our approach and to increase application and functionalities, like you making the solar cell flexible, it can be fabricated using low cost and fast methods like roll-to-roll process, and it can generate more electricity, and by reducing the absorber thickness, we can also make it semi-transparent, and the flexibility can be also good to apply it for automobile applications. And also building like new architectures is not only rectangular, like they have different shape. The flexible solar cells will be suitable for such buildings. So this was a quick overview what we are doing and what is our approach. And thank you very much for your attention. If you have any questions, please give me. And you can also write me an email. Yes.
Yes,
Thank you.
thank you very much for such interesting talk. So there is one question is asking Professor Babamurad Ahmedov, what are perspectives of solar energetics in Uzbekistan, and what is expected percentage of solar energy in Uzbekistan in next few years?
Actually, if you look at the efficiency table, like silicon, like here is a lower efficiency only for amorphous silicon. For crystalline, it's also high, but for in the market, more than 90% of solar models based on silicon, and it is cheap and compared to other technologies nowadays. but it is one disadvantage is it is sensitive for temperature. Its efficiency is decreases with increasing temperature. And as I showed, we also have outdoor performance evaluation test condition in 5 locations of Japan. One location is in our campus. So we put like several different solar panels, including like silicon-based cadmium telluride. So I think in general, all solar cells can be used, but for extreme weather conditions, one can also choose like thin film technology, which is, for example, stable for high temperature or high or low temperatures. And now this reduced price is only for solar cell, which is not included the battery, but solar energy only available in the daytime. So maybe this may raise the question: what about using the electricity during the nighttime? So we need a battery, but recently it's not shown here. For battery technology, also the price and efficiency of batteries are efficiency is significantly increasing while constantly decreasing the price. So I believe like it's not only other part or developed part countries, but also in developing countries like Uzbekistan, the solar energy has a good future and it will be available.
Okay, I don't see any other questions. So, of course, energy dependency is one of the important indicator of development of the country. I think your skills will be also very important in order to develop solar energy field in Uzbekistan. And one of the question, where could we purchase this kind of solar cells? One of the participants is asking question. Abdurrashid.
So now the last year I was
Is it available in the market?
Yes, it's available in the market, and in Uzbekistan also. Like now, I forgot the name. The small company, they are not fabricating solar cell, but they're assembling and installing to the peoples or some factories, depending on order. Like small and medium level, they're providing the solar cells and solar systems, not only solar cell, like including battery, inverter.
Okay, maybe
Yes.
if you remember the name, you may write on the chat about
Yes.
the companies. Okay, thank you very much. One more time, Abdurrashid.
You're welcome.
Now we are moving on the next presentation. Next on the line is Professor Bobomurod Akhmedov, Head of Department in the Ulubek Astronomical Institute. And Bobomurod Akhmedov is a fellow of the World Academy of Science and also a fellow of the Islamic World Academy of Sciences, editor-in-chief of the Uzbek Journal of Physics, and also editor of the Arabian Journal of Mathematics in Springer Publishing Group. The researcher of the year in 2018 in Uzbekistan, awarded by Scopus, the highly cited author of the year 2017 in Uzbekistan, awarded by Web of Science. So the title of his talk is Relativistic Astrophysics of Black Holes and Neutron Stars. Please, Professor, you may begin your presentation.
Okay. Now can you allow me to share the screen?
Yes, just a second. Now it's okay. Professor, please turn on your microphone.
Okay. Do you hear now?
Yes, now it's okay.
Okay. Let me turn to other topic, because I will speak about astrophysics. So what I would like to mention that during the last decades, there was great development of astrophysics in the world. And so it was a decade of the astrophysics, and Uzbekistan is not exception. Central Asia and especially Kazakhstan and Uzbekistan, these are two countries where astrophysics is developing very rapidly. So we have new research lines, which we did not have before. But why it is happening? Because there is a huge discovery in this area during the last years. If I would go to the Nobel Prizes, so during the last, in the 21st century, 6 awards in physics were given to the relativistic astrophysics. And for example, the last 2 years, it was continuously people from relativistic astrophysics were awarded with these prizes. So what is the triumphal now? So I would like to say that the most important discovery, it is the detection of the gravitational waves in 2016, and which was continued by the great discovery which was made in 2017 when the neutron star merger was detected. So it gave us the new era, which is called multi-messenger era. So it is one part. On other part, up to now, we did not know about the existence of the black holes. All black holes were candidates for the black hole. And in April 2019, the first image of the black hole in the center of elliptic galaxy M Messier 87 was announced. So now we are living in the era of the great discoveries for the black holes and neutron stars, which makes this area very attractive. And in our group, in Astronomical Institute in Uzbekistan, we are doing very wide research in this extensive research in this area. For example, we study optical properties of the black hole, solutions, energy extraction, electromagnetic and gravitational waves, the supernova light curves, and ionospheric study. And recently we have developed new lines of the research by new countries. And what is important in astronomy with respect to other sciences, here we used to get information about astronomical objects which are coming, passing through atmosphere. And you can see from this plot that our atmosphere is very good for the life. but it is very bad for the astronomy because it blocks the whole range. For example, highly energetic particles are totally blocked. So we have some very small window in the optical region, which is again atmosphere distorts the image of the astronomical objects, and then we can observe in the radio wave. So the cosmic era, when we put the cosmic telescopes outside the atmosphere, gave us new revolutionary discoveries. And on other hand, it is very important to have the places which with very good astroclimate conditions. And I would like to mention, from this point of view, Uzbekistan is very attractive. So the astroclimate of Uzbekistan is one of the best in the world. So we have very good seeing conditions. From this point of view, the Majdanek Observatory, which is 2,600 meters above the sea level, is becoming very important astro point in the astro site in the astronomical discoveries. So here you can see about 10 telescopes which are operating in the mount. And in addition, we have the number of other devices which we use in the Kitab station, for example, for ionospheric studies. And here you can see the distribution of the devices. And recently there is great interest from the government to develop astronomy in Uzbekistan. So you can see that new school came and new 4-meter telescope is announced to be mounted, and there is great interest to the development of astronomy in Uzbekistan due to the various conditions. And this new development of the astronomy in Uzbekistan can be done by involvement of young researchers. And if you look to the production of the young researchers which are involved in the study in relativistic astrophysics, for example, in our group, I would say that during the last decade, there were 10 PhD defenses and about 5 students, researchers who have got Doctor of Science degrees. So now we have a big number, very talented and very productive researchers who can construct new era of development of this area in our country. For example, here you can see Ahmadjon Abdjabarov. So this all Arman Tursunov and Babur Tashmatov, for example, they have made so they're the best researchers not only of our country. So this Arman and Babur were able to be best researchers of the Czech Republic. So from this point of view, the future is very promising. So we know that here you can see the number of other researchers from our group. So they are trained in the good international centers. For example, the most important here, you can see this photo of Askar and Bakhtiar Narzuloyev. So they have defended their PhD degrees in 2020 and 2021 at Fudan University in Shanghai, which is at 34th position in QS rating. And according to the decree of the president, they have got a certification for the Doctor of Science automatically, which is very, very good advantage for them. And you can see the number of other researchers. And we have our group is trying to do their best to have very good productivity in the framework of the international collaboration. Here you can see how this productivity is reflected to their, let us say, to their citations and to their other scientific parameters. And this was the part which was related to the development of this area in Uzbekistan. And the question is, what is the black hole? For example, in order to see some object, you need to get to analyze photons which are reflected from the surface of the object. But if you look to the black hole is very mathematical object. It is some point which we call singularity, which has infinite density, and it does not have size. But this object I can cover by hypothetical surface, a sphere, which I called event horizon. So in the event horizon, so even photon which penetrates event horizon can't escape. So we don't have any photons which are reflected from the event horizon, and then this object is invisible. So you may ask a question from me, why you are interested in the object which is invisible? Because even photon can not escape it, but fortunately the stars in our galaxy, they are companions of the binary systems, and in the black hole environment, the accretion disk is formed, and they, due to the highly energetic processes, they radiate in the X-ray. So in the observing X-ray binaries, we can speak about black holes. For example, there are different types of black holes. For example, astrophysical black holes, mass varies between 5 and 30 solar masses. But in the center of the galaxies, we have supermassive black holes, which mass varies between millions and 10 billion solar masses. Here we can look to the ultraviolet accretion disk. So why this process is very important? Zildovich and Salpeter independently in 1964, they have shown that the accretion of the matter, so it is falling down of the matter to the black hole, is the most energetic process in the nature. So where about 10% of the rest energy of the particles can be released. So it is huge energetics of this object, and then it is the source of the energetics of the active galactic nuclei. But then there is a theorem which says that the black hole does not have a hair. So, for example, it is means that the black hole does not have any characteristics except its mass and angular momentum. So it is the most simple mathematical objects which can exist in the nature. So it has only two parameters which astronomers are interested to measure. So if you look to any other object in the nature, it has 1 million parameters, but here it is very simple. So then the question is how to measure it. So in order to measure the mass of the black hole, it is more easy. So we can look to the motion of the stars surrounding, for example, supermassive black hole. So for this study last year, Genzel and Guess were awarded by Nobel Prizes because they have looked to the S-stars orbiting the center of our galaxy, and they have measured the mass of the central object. And so the gravity in 2000, the Gravity Consortium in 2018 measured the redshift from the S2 star, which was in the very close distance from the supermassive black hole. So it is from two parameters, one parameter is measured through Newtonian motion of the S-stars. So 50% of work can be done quite easily, but the question is how to measure the second parameter. But the second parameter is more difficult. So now for this study, we can go to the iron lines in the so now for this, we need to look to the radiation which comes from the accretion disk around the black hole. So accretion disk is very interesting because when you have the evolution of the elements, so the ferrum iron is the end of the evolution. So it is from energetic, from nuclear physics, iron is not interesting because it does not have any energy which can be extracted. And then we have majority elements which are produced as evolution of the star. It is the iron. And then now we can look to the spectrum. So iron can be excited, and it should radiate at the energy 6.3 kilo electron volt. But instead of this, we can see the widened spectra which is coming to the low energies. So these are related to the different processes, which is one of them called is Doppler beaming. So what is happening that the particles, when it is moving with the velocity of the light, almost ultra-relativistic velocities, it radiates in the direction of the motion. And then we can see that the one line becomes very, if we look to the in accretion disk, the iron atoms which are moving towards the observer, which will be much brighter with respect to the atoms which are moving in opposite direction. Here you can see it, how it looks. And then there is gravitational redshift, so which will move the particles in the low energy. So these effects can give us information about the spin of the black hole. On other hand, the inner edge of the accretion disk also depends on the rotation of the black hole. So the inner edge of the rotating black hole is 6 times less as compared to the inner edge of the non-rotating black hole. So these observations of the accretion disk show that the black holes are rapidly rotating objects. So the maximum extreme, for the extreme black hole, this parameter can be 1. And here you can see, for example, for the most famous black hole Cygnus X1, you can see this parameter is almost 1. So it means that the black holes are rapidly rotating objects. So the rotation of the object is becoming very important, and we can use it to detect for different parameters of the black holes. So I will not concentrate on this study because we don't have much time. So, but this method works only for the astrophysical black holes. But in the center of our galaxy, we have supermassive black hole. And the question is how to measure spin of the supermassive black hole in the center of our galaxy. These methods, which with iron lines and the inner edge of the accretion disk, does not work for this for the center of our galaxy. So we need to look to the other methods, and here other method is called Event Horizon Telescope, which measured first image of the black hole by in the 2019. So the question is, what is the black hole shadow? So black hole shadow is the following thing. So let us assume that there is bright star, and its image passes through the center of our galaxy. But when this image passes the center of our galaxy, the photons will be eaten by the supermassive black hole, and then we will have some hole in the image of the bright star, and the measurement of this hole can give us information about the black hole, and our task is very simple. And the shape of the this hole gives us information about the rotation of the black hole. So if black hole is non-rotating, it is a circle, but if it is rotating, we will have some dent. So there will be deflection from the circle. Okay, and but the problem is with the sensitivity, because when we look to the, we can observe shadow of 2 objects in the from the whole sky. We can observe both of them gives the number about 40 micro arc seconds. So this number is very, very small. So it is means that with Event Horizon Telescope, we can measure the size of the lemon at the surface of the moon. So you can imagine how sensitive this device, and then this sensitivity of this device is related to the size. So it is compatible with the size of the Earth. And here you can see the first beautiful image of the black hole, which was measured by the Event Horizon Telescope 2 years ago. This year there was some update also. So you can see here, it is a black hole. So we see the hole in the center of the accretion disk, and this means that the matter in the accretion disk moves on the clockwise, because when particle moves to the observers, this becomes brighter, and in opposite direction it is darker. So we can see that the motion of the matter and the shape of the supermassive, the shadow, can give us information about the rotation. And you may ask questions. So we work on this from theoretical point of view. What kind contribution which we can get through observation? Here you can see the SUFA plate, where the radio telescope RT 70 will be constructed, which will work in the sub-millimeter wavelengths. And what is important, so we made the synthetic image which can be observed by this telescope, by including SUFA telescope. And you can see that the inclusion of the SUFA telescope or the rotating black hole can give better resolution of the image, because if you look to the recent Event Horizon Telescope, it is there is nothing in our place. So now there is plan to include here Namibian telescope. So it is under construction. So it will come from the ALMA. So they are giving this test telescope, a 15 meters telescope. And if our telescope would come, so the size of the telescope will be increased, and it will increase the sensitivity of the Event Horizon Telescope. And we hope that this telescope will come in the future. So there are different methods which we can look if the black hole is electrically charged, how it may change the orbits of the stars. So can it mimic the spin of the black holes? So the answer is positive. So we can introduce other parameters of the black holes which can mimic the spin of the black hole. So from this point of view, it is becoming very important. And I would like to say that during the gravitational collapse, black hole magnetic field will be expelled. So magnetic field of black hole is equal to 0, but it can be created by accretion disk, which we observe in the observations. And then we can look how the electromagnetic field can change the motion of the test particles, which can be magnetar or white dwarf or neutron stars, which are these huge stars are becoming test particles when the central object has mass about 10 billion solar masses, and it can show us how it can mimic. So the charge of the black hole can be mimicked by other effects. And here you can see the different models for the black hole shadow. So for the standard black hole, it will be some dark hole, but depending on the parameters of black hole, it can be totally different. So it is for this, in our group, we have developed this new model which can describe the new formalism to describe black hole shadow, so I will not concentrate on it, but it gives us the whole picture of the parameters which can be measured through expansion of the different parameters in the. And other question was related to the, I think I do not have much time. Yes, Jamaluddin. Jamaluddin, hello.
Yes. Yes,
Okay.
yes.
I would like to say that
you have a couple of minutes.
A couple of minutes. So I would like to say that
Yes.
the most interesting thing, how the energy of the black hole can be extracted. So for the extraction of the energy, it is necessary that a black hole should be rapidly rotating. So if it is rapidly rotating, the ergo region will be formed from where the energy can be extracted. And then we need to look to the different mechanisms which will transform kinetic energy of rotation of the black hole into electromagnetic radiation. For this, there is Bland-Forsyth mechanism, and in our group we developed. So it was firstly it was suggested by our collaborator Naresh Dadic, and we developed the new process which is called magnetic Penrose process, which where we show that the magnetic field, even very big magnetic field, can play a role of catalysator, and it will increase the energy which can be extracted from the ergosphere. So magnetic Penrose process can be responsible for the energy extraction from the black hole and production of the high energetic particles which we can observe in the cosmic rays. So I will not, for this there is a justification which comes from the IceCube collaborations, which where it was detected that the neutrino which is coming from the supermassive black holes, so it is means that the highly energetic cosmic ray particles are produced in the supermassive black hole, which confirms our prediction. So this is what, and finally, I would like to say that from the black hole mergers, they are very interesting to study quasi-normal modes or the ring down of the black holes, which where we study the frequency and different. So here you can see that we have found a new family of the solutions in the non-linear electrodynamics, which can us give a key to distinguish is it the different types of the black holes. So you can see how the spectra differs with compared to other modes. For example, here you can see for the standard black hole, but this is for the highly the black hole in the non-linear electrodynamics. Okay, I will not speak about these things because in the second part of my talk it was related to the magnetized neutron star, pulsars and magnetars. I will not concentrate on it, so I stop here, and I would like to say that the future in this area of study is very promising. For example, here in the Astronomical Institute, we have very extensive theoretical study. On other hand, we are member of the GRANDMA project, which is looking to the electromagnetic counterparts of the black holes collisions. Unfortunately, up to now, we have about 70 events of the black hole collision, but there is no any signal which is measured on the electromagnetic signal. But for the neutron star binary merger, which was detected in 2017, the event happened in the southern hemisphere, and from that, from the optical counterpart, Chile, South Africa, and Australia have benefited. But we hope that the next merger of the neutron star with neutron star will be happened in the northern hemisphere, and Majdanek will make own contribution to it. Okay, thank you very much. Hello.
Thank you very much for this wonderful talk, Professor. There is one question for you. So, Professor Abror Gadayev from the National Center at Samarkand State Architectural and Civil Engineering Institute asking a question. So, how do you think, what is the relation between black hole condition and climate change in Uzbekistan? Because it's somehow related to the water resources. And next question is, did you interested or investigated water resources and black hole condition changes in Central Asia? Thank you in advance.
Okay, good question. But I think we are happy that we are in the peripheral part of our galaxy. So we are very, very far from this extreme conditions. So you see the frequency of the gravitational wave frequency, it is 300 hertz. So it is means that in 1 second, you need to make jumps 300 times. I think everything will be destroyed. So we are happy that we are very, very far from black holes, and then we are in the quiet place of our galaxy. So we are very far from it. And our sun is also a small object. So if you look to the mass of the stars, so they can go up to 200 solar masses, which are 200 times more massive as compared to our sun, and they're more catastrophic. But our sun is very good, very quiet, and long-living object. So it lives almost forever. From this point of view, we are very lucky. So we have almost infinite time for civilization, for other things, but the climate change is made by human. We need to afraid human, not black hole. Human who produce a lot of carbon into the atmosphere, and this carbon will change. Of course, his question is very important because I think the important thing, so we need to have more water. Of course, water is most important for the life. And when we look to the, you see there are about 5,000 exoplanets observed in our galaxy. So almost each star have about 10 exoplanets. So planetary system is very common in our solar system. And then we look to the existence of the conditions for the life in the planets, and now we have a few dozens of the candidates for the habitable planets. So there are conditions, including water, in that planets where the life can exist. Okay, this is these questions are very interesting. So we hope that okay.
Thank you.
But
Thank
we
you,
are
Professor.
lucky far from the black holes. Okay.
Thank you for your answer. It's cosmology, black holes. It's always interesting side of the physics, and for me,
no, this I say that astronomy in our blood.
Yes.
So, for example,
Yes.
if you, I say we are from desert. So when we go to a desert place, we look to the sky and we see all stars, and then we saw it for many generations. And from this point of view, astronomy is the most interesting subject in our region. So
Yes, indeed.
I'm sure for this. And then since we have very good astroclimate, we need to develop astronomy in Central Asia.
Yes, yes.
Okay.
Thank you very much one more time,
Thank you.
Professor, for your interesting talk. Now we will move to other speakers. So following speaker is Dr. Siraj ul Haq. He is an assistant professor in the Department of Chemistry at University of Azad Jammu and Kashmir Muzaffarabad in Pakistan. So he mainly works on synthesis of characterization of nanomaterials and their evaluation for environmental application, like absorption and photocatalysis, and biological applications like antibacterial activity and antioxidant activity. So please, Dr. Sirajul Haq, you may start your presentation. My apology, I don't see Dr. Sirajul Haq in a list of participants. So in this case, I'm gonna move on to the next speaker. So our next speaker is Dr. Umidjon Khalilov. He's a leading researcher at Institute of Ion Plasma and Laser Technologies in Uzbekistan, and he's also participates in investigations that taking place in University of Antwerp. Dr. Umidjon Khalilov obtained his PhD in 2013 at University of Antwerp, and he won one of the prestigious fellowship of FW twice in Belgium. So the title of his talk is Nanostructure Synthesis Inside the Carbon Nanotube. Please, Dr. Umidjon.
Assalamu alaikum. Thank you, Kamal, for the invitation. Also the organizers for the giving opportunity to give the present my recent results. And today I will try to introduce small introduction about carbon nano technology. So, so the title, the nanostructure synthesis inside of carbon nanotube, and you know, everyone knows about the silicon technology. So silicon technology miniaturized electronic devices and made them powerful. You know the silicon technology because of your cell phone, your TV, or more things, electronic devices. However, the silicon nanotechnology is being considered, in the recent year, its effectiveness has declined sharply because of the microchip reached their physical limits of miniaturization. Consequently, carbon nanotubes, carbon-based nanotechnology is being considered as a viable alternative to the carbon nanotechnology being used in electronics, materials, material science, or biotechnology, nanotechnology, or energy saving. Everywhere is used. Carbon nanotechnology is based on the manufacturing of the carbon nanostructures. Here you can see many of the nanostructures. Some of them you know, for example, fullerene, nanotube, or graphene, this kind of structures you know already. So carbon nanotechnology is based on the manufacturing this kind of nanostructures. These nanostructures that have how to say, extraordinary properties, which strongly depends on their geometry. Therefore, the structure synthesis is challenging issue in the nanotechnology. In this talk, I would like to say about one of the options for obtaining nanostructures with desired geometry is individual synthesis. Here you can see carbon pivot. So this is the carbon nanotube inside fullerenes. We can see in this schematics diagram the non-catalytic synthesis of carbon nanotubes in parent tube. The various precursors here first encapsulated into the tube and then subsequently annealed to induce the growth, and then inner tube is extracted by sonication treatment. Other very good carbon nanostructure is graphene nanoribbon. The fabrication of graphene nanoribbon is also important area to investigate, since the band gap increases with decreasing the width of graphene nanoribbon. However, this method uses more expensive precursors. You can here see the preparation of these precursors are also the expensive. That's why I would say this is not so cheap experiment. That's why I investigate. We can use more simple precursors. By using these precursors, we can get this kind of structures. And I investigate this problem using a computer experiment. So computer experiment is the powerful tool to describe real experiment or physical processes or chemical processes, including intermediate processes, which experiment can't reach sometimes, can explain some nanoscale processes. And that's why computer experiment is nowadays using widely together with real experiment to explain some experimental results. In the computer experiment, we used different simulation techniques, methods, and then for this, using these methods, we try to explain or understand more physical and chemical processes. In that case, for the performing computer experiment, we need supercomputers. Here you can see huge of the computer processes together joining the one place. This kind of high power, due to this high power supercomputer facility, we can perform computer experiments. Let me introduce some of my results related to inside the nanostructure growth inside nanotubes. For the simulation, we got this kind of nanotube, and then inside we inserted nickel catalyst, and then carbon or hydrocarbon gas subsequently it's inserted into a nanotube. Then what happens? We can see here, if we use this precursor, so carbon precursor, we can get this kind of metal containing fullerenes or nanotubes or carbon, new type of nanostructures. On the other hand, if we use hydrocarbons, we can get nanoribbons and bilayer graphene nanoribbons. What's the reason? The reason is the competition between catalyst and feedstock. Due to this competition, during the dimerization chain, ring, and the cap sheet formation steps, the either grows this kind of cap or this kind of sheet as the initial structures. Then we can get this kind of nanotubes or this kind of graphene sheets, nanoribbons. We try to compare our results, obtain results with experiment. So you can see the diameter difference between the outer and inner wall, the obtained double wall carbon nanotube. You can see 3.3 angstrom, which is in the experimental observation. It corresponds to experimental observation. Also graphene nanoribbons, also the width of graphene nanoribbons exactly corresponds to experimental observations and results. Also, we check the effect of catalyst, size of catalyst. We found that almost no effect the size of catalyst. It means if we use 5 atoms catalyst or 18 atoms catalyst, the quality of appearing this kind of nanostructures are almost same. Overall, the results indicate that the structure control highly depends on the choice of feedstock in this catalyst assisted flow. To evaluate the stability of these obtained structures, here you can see 5 kinds of structures. So double wall carbon nanotube, and carbine in double wall carbon nanotube, graphene nanoribbon inside single wall carbon nanotube, hydrogenated and double hydrogenated single and double layered graphene nanoribbons inside single wall nanotube, as well as their the metal-free counterparts. You can see we try to estimate the stability, relative stability of those structures using this formula, which is called a molar Gibbs-free energy of formation. Then the green one related to this and the red one related to this. You can clearly see that the stability of the metal-free structures are more stable than metal-containing structures. That's why we conclude that we should withdraw these metal atoms from inside of the nanostructures. Then consequently, the oxidation-based purification has been studied. During the oxidation process. You can see the concentration of nickel atoms decrease step by step. While oxidation slightly damages outer tube, parent tube, you can see the quality is a little bit changed because of the defect creation. The inner tube retains quality. Here you can see. Such oxygen-based purification of metal-containing double-wall carbon nanotubes have already experimentally reported in the grow double-wall carbon nanotubes from metal assays. However, the purification mechanism has not been properly explained. That's why we try to explain the purification mechanism. Purification mechanism consists of the 2 stages. First stage is defect hole created here. You can see when an oxygen atom detached carbon atoms from here, and then subsequently plugged nickel atoms gradually detached from the tube. This process cycles until the tube releases anhydral metal atoms. One of the very novel structure, carbon nanostructure, is a carbine. Carbine is the linear chain. It consists of linear chain of carbon atoms. Carbon has extraordinary properties. Specifically, you can see here that its mechanical properties significantly outperforms any other materials, including carbon nanotubes and graphene. However, the synthesis of long carbon is extremely difficult because of its high chemical reactivity and instability. Therefore, the inner space of the carbon nanotube has been recognized as an ideal location for the carbon growth. Consequently, record long carbon in the double wall carbon nanotubes, you can see, have experimentally reported. However, despite of such valuable experimental theoretical investigations, effect of feedstock as well as the role of the feedstock catalyst tandem in the endohedral carbon growth is not reported yet. Also, the growth mechanism is not properly understood. That's why we try to analyze this process, growth process, using computer experiment. For for our computer experiment, we've chosen the double wall carbon nanotubes 5, 5, 10, 10, and inserted some of nickel atoms, nickel cluster, we can say, and then use 3 kind of feedstocks. Here you can see the effect of feedstock. In first case, we have grown the carbon. Here you can see carbon. If we include hydrogen, or the feedstock consists of hydrogen atoms also, we can see not real carbon, it's like hydrogenated carbon. If the hydrogen concentration is increased, you can see here the more disruptive structures. Here you can see the nucleation of growth tips of nickel catalyzed in the hydrocarbon. It's really really depends on the feedstock. Namely, the catalyst facilitates here this the processes. Hydrogen has opposite effect during the growth process. Namely, the catalyst facilitates the formation of carbon-carbon bonds here. However, the hydrogens and hydrogen prevents this kind of transformations. In particular, molecular dynamics NIP calculation also performed to show the formation of initial carbon chains from the feedstock dimerization recorded, which you can see. It depends on the association activation barriers. Association activation barriers depends on the feedstock related to growth. And also the length of the carbon is related to the hydrocarbon species. In overall, catalyzed carbon growth depends on the catalyst activity and hydrogenation degree, you can see. And due to this activation, we obtained different kind of structures, carbon structures. Here you can see it's polyene or polyene, cumulene, and polyene chains, or the polyene chains like this, which is experimental, also obtained this kind of structures. Here you can see also. Consequently, the connection of catalyst and then hydrogen to the chain, carbon chain, can affect both its length and then stability. Such additions don't only alter the morphology of the structure, they can change also the electronic structure, properties of obtained carbon. For example, this figure demonstrates that the overall gap width significantly decreases when the hydrogen atom added along the nickel termination carbon chain. If you compare these numbers, the structure, you put here the structures, 1, 4, 7, 12 structures, you can see we added hydrogen concentration, we increased the hydrogen concentration this way, and we got the band gap decreasing. So it means this structure, semiconductor, if we hydrogenated this structure, it becomes like metal. It's like it's not anymore semiconductor. Also, we also found the current in the functionalization samples becomes larger than the obtained pristine carbon. Here you can see the larger current here in the hydrogenated carbon, just comparing with pure carbon. So it means the hydrogenation or chemical modification considerably changed the electronic properties of obtained carbon. Let me conclude or summarize all of the results. So the selective growth of encapsulated and the methylated tube or ribbon is respectively determined by a catalyst or feedstock domination in their computation. Methylated encapsulated carbon nanostructures were found less stable than metal-free carbon nanostructures. Consequently, oxidation can assist to purify encapsulated carbon structure from metal atoms in order to obtain stable counterpart. The electron structure of nickel terminated carbide can be tuned by steering the hydrogen concentration along the chain. In particular, increase in the hydrogen concentration leads to a decrease in the band gap of carbide, which shown in the previous slide. At final, I would like to thank my collaborators and also Foundation, FW, to help to all of the supporters, different kinds of supporters. And also thank you for your kind attention.
Thank you very much for such an interesting and informative talk, Dr. Umirjon Khalilov. If you have questions, please write on the chat. Currently, I don't see any questions, but I have one. So according to your research, if the radius of the carbon nanotube is small, the inserted nanoparticle leads to the growth of carbine, right?
Yes.
If we increase the radius of the carbon nanotube, so different kind of nanostructures might form, if I understand correctly.
Right.
So it's a matter of size and
yes.
number of particles inside of the carbon.
Yes, there are many parameters for to grow or to control this kind of growth. For example, one of parameters is size of the or diameter of the carbon nanotubes. Also the type of the feedstock here you can see, and then type of the catalyst also we can see, and also the temperature is effect there are, and also the pressure effect of the feedstock. There are many effects. We should carefully choose those parameters, and we then get this kind of structures.
Okay. It's a quite complicated process. Yes. Carbon nanotube and
yes,
carbon.
I introduce it, but there are many specific things there.
As far as I know, if someone finds a way growing the 100%, for example, carbon nanotube, it would probably lead to the Nobel Prize, right?
Exactly. If we learn how to control the structure, we can get Nobel. But it's really difficult. It's really difficult. Not so easy.
That's a fundamental basis of growing carbon nanotubes by nickel nanoparticles. I think in a near future, human race will obtain how to synthesize 100%, for example, chiral or armchair, or what was the third one? I don't remember.
Graphene nanoribbon. Yeah.
Nanoribbons as well, right? If you there is no any other questions, thank you for your participation, Dr. Umirjon Khalilov. So due to the technical reasons, Dr. Sirajul Haq cannot connect at the moment. Now he is online. Let me introduce Dr. Sirajul Haq one more time. So he's an assistant professor in the Department of Chemistry at University of Azad Jammu and Kashmir, Muzaffarabad in Pakistan. He mainly works on synthesis and characterization of nanomaterials and their evaluation for environmental application like absorption and photocatalysis, and biological applications like antibacterial activity and antioxidant activity. So please, Dr. Sirajul Haq, you may start your presentation. please turn on your microphone. Just a second.
Jamal'ın tugattınız mı?
Hazır. Co-hostlarım member nimani
It's a notification that the host is not allowing you to unmute. So please. Okay.
Yes. Okay. Dr. Surajul Haq, you can start your presentation.
Assalamu alaikum and good evening to everyone. Sorry for this little interruption. I'm Dr. Siraj and I'm going to deliver my talk on the plant-mediated synthesis and characterization of biometallic nanocatalyst, biometallic nanomaterial. First, the presentation overview is the introduction to the nanomaterials, synthetic approaches used for the synthesis of nanomaterial. And then we will emphasize our talk on the green synthesis, mean the plant-mediated synthesis of nanomaterial. Then we will take an example for the preparation of biometallic nanomaterial, and then we will know about the characterization technique used for the characterization of that nanomaterial. So basically, we have three types of particles. That's coarse particle, fine particle, and nanoparticle. And this all depend upon the size parameter. On the basis of size, we have divided the particle and three.
Sorry, Siraj, could you share
Yes.
your screen, please? Now, it seems our administrator made you a co-host. Could you share your screen and keep talking, demonstrating your presentation, please?
It's okay now?
Now it's okay, yes.
Yeah. Oh, sorry. So we have the particle and the basis of size can be divided into three types. That's coarse particle, and these are the particle whose size are from 2.5 to 10 micrometers, like fallen grains or dust particle. And the second one is the fine particle, and the size of these particle are 0.1 to 2.5 micrometer. Secondly, the third one is the nanoparticle, and nanoparticle are defined as the material that have 1 to to 100 nanometer, or at least in one dimension. The particle that have size between 1 to 100 nanometer and at least in one dimension. Recently, some scientists, researchers believe that for industrial scale, the nanoparticle, the size of nanoparticle should be between 1 to 1000 nanometer. But conventionally, we are going in the conventional definition that nanomaterial should be in the size range between 1 to 100 nanometer. So bimetallic nanocomposite, bimetallic nanomaterial, bimetallic nanoparticle. Why we are emphasizing nowadays on the bimetallic nanoparticles? So one of the basic reason is that large number of research has been done on the unimetallic material like zinc oxide, tin oxide, titanium oxide. So large work has been done on these type of materials. So now we need some more advancement and some new properties. So in search of these properties, to enhance these properties of the nanomaterial nanoparticles, we are going to synthesize the biometallic nanomaterial. Or the first thing is the very limited data is available on the synthesis and characterization and application of these material. The coupling activity of surface site for multiple application. These nanoparticles exhibit the novel properties of multi-component system in the same material, and novelty expected due to the synergistic effect of the counterparts. So the synthetic approaches for the nanomaterial. So basically we have two type of approaches. That's top down and bottom up approach. And these two are totally different approaches used for the synthesis of the nanomaterial. Basically the top down approaches, there's a physical method, physical synthesis of the nanomaterial, while the bottom up approaches that are the chemical synthesis or chemical methods used for the synthesis of the nanomaterial. So these are some physical method. There's a ball milling method, high energy ball milling method, physical vapor deposition, thermal evaporation technique, and sputtering method. In this method, in top-down method, we convert the bulk material into the nanomaterial. And these 3-4 process, that's grinding and cutting and vaporization, or the thin process. Either we can grind the bulk material to convert into a nanoparticle, or we are going to cut that bulk material to convert them into the nanomaterial, or we can vaporize and deposit this, it on a surface. So these are the 3 processes used during the physical synthesis of the nanomaterial. And these methods have some advantages that we can use these methods for the large production of the nanomaterial. And these methods need no chemical fabrication method, no chemical fabrication is needed. Here is some limitation that it's a broad size range, that's from 10 to 1000 nanometer. The particle synthesized by this method, the size and shape are not uniform. They are highly expensive methods and high surface defect. The particle synthesized by this method are highly they have high surface defect. Some other limitation is also not noted here. That's in this technique, we can only synthesize the metal nanoparticle. Let's suppose we are going to synthesize the copper nanoparticle. So we need a copper sheet. And then we are going to process that cutting, or grinding, or vaporization process. And then it will be converted into the nanoparticle. But if we going to synthesize, let's suppose copper sulfate or copper sulfide, copper oxide nanoparticle. So these methods are not valid for the synthesis of derivatives of that metal nanoparticle. It means metal oxide, metal nitride, metal sulfide. Only the vapor deposition method is used for the synthesis of the metal oxide, because we can vaporize the that bulk material in the oxygen environment, or we can vaporize in any other environment. We are going to synthesize that type of material. But usually these physical methods are used for the synthesis of metallic nanomaterial. Similarly, another type is chemical synthesis. So sol-gel metal, colloidal metal, chemical precipitation metal, microwave radiation, sonochemical reduction, and chemical vapor deposition metal. These are the metal, and some metal also beside these metal used for the chemical synthesis of nanomaterial. In this process, we are going to ionize a precursor salt. Let's suppose we are going to prepare copper oxide or copper nanoparticle. So first we take a precursor salt and dissolve that in a solution, in a solvent. And after ionization, the nucleation process starts. And after nucleation, the growth process occurs. So this is the bottom of way for the synthesis of the nanomaterial. This method have some advantages. That is like ultrafine nanoparticle production. This metal is economical as compared to the physical metals. The particle form on this metal are uniform in size and shape, and the growth of nanoparticle can be controlled. Disadvantages or large-scale production is difficult. The chemical purification is needed, and high surface defect. Some of the particle have high surface. Sorry, it's a low surface defect of these particles. In chemical synthesis, these methods provide us the opportunity to synthesize various derivatives. Let's suppose copper nanoparticle. We can synthesize copper nanoparticle in this method. We can synthesize copper oxide nanoparticle using this method. We can synthesize copper sulfide method. Or copper sulfide using this method. We can synthesize copper nitride using this method, and all these pyramid. We can set parameter for this synthesis. Means if we are going to synthesize oxide, so we need oxygen environment to synthesize copper oxide nanoparticle. Beside these two metals, this green synthesis has been recently a decade before was introduced, and this process we are using the green sources, means bacteria, fungi, and plant material. So mainly we will emphasize on the plant synthesis, or green synthesis of the nanomaterial. Why we use plant extract for the synthesis of the nanomaterial? Because it's fast and single step method, it's cost effective, it's environment and health friendly, because in this method we didn't use any toxic chemical like sodium borohydrate, citric acid, and etc. We need no high pressure, temperature, and toxic chemical in this metal. And this metal is easily scalable for the large scale. Now, how we can use plant material for the synthesis of the nanomaterial? For this purpose, first we going to select the plant material. Plants, first we had to done one thing. First, to study the phytochemicals of that plant material. A large variety of literature is available. More than 80% plant for the more than 80% plant phytochemical studies are available in literature. So first we can find that what type of phytochemical present in that plant, because we need such chemicals, such natural product that possess OH group, hydroxyl group, because that hydroxyl containing compound is a reducing and capping agent during the synthesis of the nanomaterial. Then after that, we choose the pot, that which part of the plant we are going to use during the synthesis. We can use roots, stems, fruit, and leaves. But the best way to use the extra pot, means if we are cooking vegetable in home, so that's extra pot we are going to dispose. Let's suppose we are going to cook potatoes in the home, so the potato peel we can use for the synthesis of nanomaterial, like orange peel, like watermelon peel. We can use that's extra material there we are going to dispose, so we can use that for the synthesis of the nanomaterial. And for the typical synthesis, let's suppose we take 50 grams of that plant material, plant material, and we can dissolve, or we can add it into the 100 ml distilled water and boil for 3 hours. So after 3 hours, we obtain a dense color of the cloud extract. This cloud extract can be cooled down at room temperature, and then after that it should be filtered, and after filtration it should be centrifuged, and after centrifugation it should be stored below 10 degrees C in an airtight jar, because this is the favorable growth for the growth of fungi. So after week or 10 days, the fungus grow on this extract. So we should be very careful during the storage of the these extract. This extract is acting as reducing and capping agent. So this extract replacing the reducing agent like sodium borohydrate or some polymer we are using is a stabilizing and capping agent for during the synthesis of nanomaterial. So this extract eliminate, this extract replace the use of the toxic chemical during the synthesis of the nanomaterial. Now, let's suppose we are going to synthesize zinc cobaltate bimetallic nanomaterial. Zinc cobaltate is a bimetallic nanomaterial, and we are going to synthesize that nanomaterial. So zinc cobaltate containing 2 metals. That's one is zinc, another is cobalt. So first we are going to take the precursor salt, that's sodium or zinc sulfate, whatever the salt, that's maybe chloride, that may be sulfide, that may be acetate, that may be nitrate. So first we are going to prepare a solution of the precursor salt, and then we are mix this precursor salt solution with the plant leaves extract. So the ratio we have take here is no matter that what you are going to take the ratio. That's maybe I have take 50 ml of the zinc sulfate and 50 ml of the cobalt nitrate solution, and then mix that with the 20 ml of the plant leaves extract I have prepared previously. Then by combining these two solution, we are going to heat and stir these for 1 hour. And after 1 hour, we see that the gel formation occurs. The gel is basically the hydroxyl network of the metal hydroxide network. So then after the formation of the metal hydroxide, we going to wash that metal hydroxide to remove the unreacted salt and other ions. And after that, we going to mix that two hydroxides and stir and heat it for 4 hours. After 4 hour heating and stirring, we age this reaction mixture for 24 hours. And after 24 hours, we can wash and dry the solid product and dry it in the oven. After the preparation of this solid particle, it can be submitted for the characterization, and it can be used for the different application like as a catalyst during the organic synthesis. It can be used as a photocatalyst for the degradation of toxic pollutants. It can be used as a resorbent for the absorption of heavy metals and toxic dyes. It can be used as antimicrobial agent and antioxidant agent for means is could be used for different biological processes. Even we can use it for as a drug delivery agent. So this is the simplest way which replacing the chemical method in which we use a high toxic chemicals, and also replacing the physical method which are highly expensive methods. The nanoparticle form on this why this method are mostly have uniform size and high surface area and uniform shape. These nanoparticles then can be characterized to study their crystalline nature via XRD, FTIR, surface area analysis will be carried out of these nanoparticles. The scanning electron microscopy, the transmission electron microscopy, energy dispersive X-rays, diffuse reflectance spectroscopy, and thermogravimetric analysis. So the XRD is used to study the crystalline nature of the material, that whether the material is crystalline or amorphous. And then we can calculate the crystallite size and other crystallitic parameters. The FTIR is used to study the functional group, to study the band, the bond, the stretching and bending modes of the material. The surface area is analysis. We are mostly using nitrogen absorption desorption technique. So we can calculate the surface area and pore size and pore volume of the nanomaterial. The scan SEM and TM are used for the to study the morphological appearance of the nanomaterial. The ADX is used for the elemental analysis, where the diffuse reflection spectroscopy is used to calculate the band gap energy of that material. And TGA is used for to study the thermal stability of the nanomaterial. So these the green method is recently is a very efficient method for the synthesis of the nanomaterial. It's a very low cost and very efficient method, and it's the most important. It's eco-friendly and user friendly. Thank you.
Thank you very much for your interesting talk, Dr. Sirajul Haq. So, do we have questions? So, I don't see any questions, but I have. Yes, Umeed, go ahead.
Yeah, Umeed.
Assalamu alaikum, brother. How are you?
Walaikum salam, brother. Thank you. Fine.
Thank you.
How are you?
Fine. So,
Yes.
Very good talk. Thank you for this kind of information, because it's useful for us. I'm also doing the making catalyst by computer simulations.
Yeah.
So, my question is, can we use your extracted nanoparticles for the growth of carbon nanostructures?
It's not reported yet.
Not yet.
Not yet. It's not reported yet. I have not studied it, that the carbon nanotube can be synthesized by this metal. One thing I have studied last week, that the graphene can be synthesized via using this method.
Really?
Yes.
Then we will talk later.
Yeah, of course.
Okay. Thank you.
Oh, welcome.
Okay. So, Dr. Sirajul Haq, is it possible to do massive production of catalysts by means of plants? And is it financially beneficial?
Yes, of course, it's most beneficial, because we need only one precursor salt. Let's suppose I am going to synthesize the zinc oxide nanoparticle, zinc and cobalt bimetallic nanoparticle. So I need just precursor salt. Nothing, no reducing agent, no stabilizing agent, no any other thing. And a very moderate temperature, that's 50 degrees C. And distilled water. We need a hot plate, one beaker, magnetic stirrer, distilled water, plant material, and precursor salt. So I think this is the cheapest way for the synthesis of the nanomaterial.
Okay, thank you. It's quite interesting.
Welcome.
Because it's catalyst is most required. It's nanoparticles and production around the world. I think obtaining where this kind of catalysis cheap in price, it would be very, very beneficial, yes, and profitable business, yes. Thank you very much one more time for your
welcome.
Now, on the next line is Dr. Talant Ruzametov. So Talant Ruzametov obtained his master's degree at Mississippi University in 2010, and then he also obtained his PhD degree at Kent State University in Ohio, USA in 2019. Now he's working as a senior researcher at Rutgers University in New Jersey, USA. Welcome, Dr. Talant Ruzametov. You may start your presentation, please.
Hi, thank you, Jamal. Can you hear me?
Mm-hmm.
Okay, so it's really early morning here, so I'm gonna keep my camera off if you don't mind.
Mm-hmm.
So
okay, so could you share your slides as well?
Just a second. Do you see it?
Yes, it's okay now. Go ahead.
Okay, so Assalamu alaikum, everybody. So I'm really delighted to give a presentation in this conference. So we go long back with Jamal. We did undergrad together, and I'm also very happy to see some of my teachers and some of my classmates in the group. So thank you. So I'm going to start with my title, Understanding of Biomolecular Interactions with Deep Learning. And so I got into this kind of research during my postdoc at Lamoureux Lab at Rutgers Camden. So I work at this center called Center for Computational and Integrative Biology. And the campus is located near the Philadelphia. So we have big universities around us like Penn State, UPenn, Princeton, and Rutgers. So a little bit about research overview. So the long-term goal of the lab is studying the protein-protein interactions, protein folding and binding, using deep learning. And the ideas we derive from deep learning are such as end-to-end differentiable learning, representation learning, energy-based models, and we also participate in CASP computation. So end-to-end differentiable learning is the terminology that refers to deep learning. For example, already in deep learning, everything is differentiable, meaning the network is differentiable with respect to all the parameters. But later, it got more and more complicated, and the society, they started putting ideas from physics into it and make those things differentiable. So that's why this terminology arose. Now, this energy-based models is one of the examples on that. For example, the representations that's generated by a neural network, let's say convolution neural network, they can be correlated with each other, and then the product can be interpreted as kind of pseudo energy. So today I'll be mainly focusing on two projects, although there's many more. So one of them would be this small fragment free energy map prediction using AC3 convolution neural networks. So this project is in collaboration with Alexander Makarov from University of Maryland and Silks Biopharmaceutical. So, and then the second project I'll talk about would be about learning latent representations of tripeptides, these small molecules. And both of these projects are actually aimed to alleviate problems in MD simulations because they're too much time consuming. So that's one of the main goals. So the second project is in collaboration with another pharmaceutical company from Canada. Okay, so here I have a very simple objective of how these deep learning models are formulated. So if you want to solve any problem, we represent that we need the data that represents the problem, and then a label associated with that. So data is labeled as X here, and then the label would be Y prime. And then when we, during the training, we give that data into this neural network. So neural network is like a black box. So we give data as an input, and then P is going to be parameters. So it can have a lot of parameters that's going to be optimized during the training, and F represents neural network. And then at the end, the output that comes out of the neural network which is Y here. And then that Y is going to be minimized using loss function. So we simply can take the difference between Y and Y prime and minimize the mean square loss. And then we train for some time, and then we optimize the parameters of the network, and then that can be used to predict new labels for unseen data. So that's the very simple interpretation of all these predictive models. Now, this neural network can be as simple as multi-layer perceptron. This is one of the very initial types of neural network. And then convolution neural network, which kind of revolutionized this field. And then GANs, and then later these transformers are famous now because of this their usage in AlphaFold prediction of this 50-year-old protein folding challenge. Okay, so here I'm going to introduce some basic concepts about convolution neural networks, because that's what we have been mainly using for both of these projects. So the convolution operation is a very simple operation. Actually, it's just it's composed of multiplications and additions. It's not the actual convolution we use in physics. The mathematical term would be correlation, but they somehow adopted this term because it sounds more fancy, I guess. So the way it works is, so if I have this image like embedded as ones and zeros, and this is supposed to be moving, I guess. This window can slide over the image. So this large one is the image here, and then this window can slide with some stride, and every time it's sliding, it performs element-wise multiplication in addition, and then whatever it gets, it puts in the middle. And then this is a new feature coming out of the network and getting passed to the next layer. So here, this operation gives me 4, and then we can slide further and then get this representation and pass to the next layer. So that's convolved feature. Now, this operation, as you can see, is very linear, and none of the problems we work with in real life, none of them are linear. They are non-linear. So that's why we often apply non-linearity functions at the end of the convolution operation. And basic examples would be sigmoid. They tend to suffer vanishing gradient problem. That's why the better method is value. And this is how it looks functionally. Now, one main problem with this convolution neural networks is so-called rotational equivariance. What do we mean by that? For example, let's say I'm trying to learn the image of the cat. So I give a lot of images of the cat to my network, and I'm trying to classify from the images if there is a cat or if it's something else. So often these constnets, they can, for example, learn the translated images. For example, if this is the frame I'm passing to the network and there's a cat image, I can put it in any place in the same orientation, and network can perfectly identify a cat. But instead, if I rotate that cat image in the input a little bit, it's not able to identify it because it doesn't have a property called rotational equivariance. Now, the term I'm using as equivariance is usually when quantities are scalar quantities, we use the term invariance. here we use equivariance because sometimes the method can have vector features and they need equivariance because vectors they rotate locally and then globally. So the operation is shown in this formula here. For example, if F is the neural network and G is the group operation, so it's like a SO3 rotation operation. So I can apply that operation on data and then give it to network, and then the output should be same as applying that group operation into the network after I give it data. So that's the basic concept. Now, here is more better visual illustration of that concept using 3D protein structure. So let me play that. So as we can see here, I'm giving that protein structure as a density into my model, the neural network model, and then I rotate the input, and if the model is equivariant, I expect the output rotate in the same way. And as an output here in the middle, I show a vector feature. So it's a feature with L1. The L1 refers to the first order of spherical harmonics, and this is scalar feature. So these vectors have both local and global in equivariance, and we use this repo for doing equivariant neural networks. Okay, let's move on. Okay, now moving on to the projects I mentioned about. So the first project is called learning fragment-based prediction of the fragment-based drug design frag maps, basically. So in this project, so we have this collaboration with SilksBio, and then they have a method called Silks, where they do molecular dynamics simulations to find the free energy maps of small fragments binding to the proteins. So this is the field is called fragment-based drug design. Basically, after generating this kind of frag maps, they use it to predict ligand binding free energies, which is very essential in drug design, because if you want to design a drug, there are so many small molecules, and to sort out which molecule works best for some specific drug. The computational methods are the best to select a few from the candidates, and it saves a lot of time because time is important in this industry. So let's see. So what they do is basically they choose small molecular fragments based on their the chemical group properties, such as these, and they are labeled and color coded here. And then they run MD simulations and they generate equilibrium densities and free energy maps. And then that method is better than experimental method because it takes much less time, but still it takes on average like 4 days to generate those structures per protein. Now, we had an idea to reduce that time into 1 second, and we actually achieved that, which is really efficient. So we did that with deep learning. So I'm going to talk about that in next slide. So how we did that. So we used convolution neural networks, which is AC3 equivariant. So we put protein structure as a density into 11 grids labeled based on their protein side chain groups, with these groups here labeled. And so we pass that into network, and we train that with that network. And at the end output of the network, we minimize based on these moieties of the functional groups, and this data is provided by SilksBio. So they give us free energies on a grid for each this functional group, and then we use that to minimize the loss function. So, and here are the sum of the results with that. So we use this with spherical harmonics order 2. So why do we use spherical harmonics here? Because that idea is used to construct this AC3 equivariant filters. So let me go back here. So So usually when these neural networks are not rotationally equivariant or invariant, the common practice is called rotational augmentation. So the input is augmented by doing rotations, and that's very costly operation. So this AC3 equivariant networks kind of very helpful to overcome that. So instead of doing rotation in the input, what they do is they change the filters and then do rotation internally in the network, which is much more efficient and better. So now back to the result here. So now here I show the results using model above here, and this should be rotating. Okay, sorry for the technical problems here. It's okay. So what I show is here is the prediction result for this protein Y1YCR, which is egg glycoside protein, using L2 order. So L2 means there's a scalar features, order 1, vector features, order 2, and tensor features. And the results are shown for the free energy iso values of these, as shown for generic apolar, methyl ammonium nitrogen, and acetate oxygen. And as you can see, the original data is labeled in green, and then the brown shows the predicted results. And here we also show the ground truths versus the prediction. As we can see, these results are not perfect because while doing this project, we realized that we need more data, and then this became a grant proposal. So, and it's still an ongoing project. Now, moving on to the other project, which is again, we use MD-based data to learn the representation of the tripeptide. So what is this about? So there is an famous idea, I think everybody's familiar with this, it's called deepfake. For example, if you guys have seen model which imitate the facial expressions of the people, so it's called deepfake. So we derive those ideas and use it for the good cause, meaning to learn conformations of these small protein structures, specifically this tripeptide. For example, here I show a video for like conformational sampling using MD for a tripeptide composed of alanine, alanine, alanine. And so in order to learn how this conformation changes, that's really important, because once we learn it, we can generate those structures, and also we can compress this into a latent one-dimensional representation. So we are given this MD data from our collaborator. It's called Procure, pharmaceutical company. For example, for this specific training, I'm going to show, so we used like 1,000 conformations per tripeptide, and those trainings were done on AXA, X meaning all amino acids, and at the endpoints, they have alanine attached only. And these MD sampling is done using replica exchange molecular dynamics. So the network, one of the network types I show here is a simple autoencoder, where we start with 11 densities mapped into the grid, and then we slowly reduce that into one feature, and we keep the spatial dimension same. So, and then at the end, the output we minimize. So the target is going to be the same input. So this means this is unsupervised learning, and we are just compressing the data. Now here I show some of the results. So here's the loss function of the training and validation. So for the validation, we use unseen conformations of the tripeptide. So here what I'm showing is so trained model, and once we do the training, the new unseen confirmations can be passed to the network, and then I'm showing the input, latent feature, and the output. As you can see, output reconstruction is nearly good, but it's a bit hard to derive meaning from latent representation. But what we can see is it's basically tracking some of the edge motions. It could be the backbone or locations of the groups. So this is a big field now. This learning latent representations, and it's usually hard to interpret this, the meaning of it, because what's going on inside the neural network is itself hard to interpret. So let's move on to the next slide. Now, what's more interesting about this specific type of architecture is, so during the training, we can use a small grid size, meaning the number of voxels are small, like 20 by 20 by 20. But then that learned representations can be used to derive representations for larger structures. For example, I can use that model and give my protein, like any protein of any size, and it can do the perfect, nearly perfect reconstruction. And that's one of the ideas in representation learning. Like you learn from something smaller, and then you transfer that learning into something different. Could be something bigger. So that's one of the useful aspects of this project. So here I show that example for this one YCR, which is equilisozyme. So another idea I show here and represent the result is for the interpolation, linear interpolation. For example, what I show here is, so we can take a structure of this tripeptide, and then there are different conformations. And what we did is we took we generated a different conformation from this Rotamer database, which makes sure that this rotations of the side chains are proper. And then one can do the input this initial structure, and then this structure, and then we have two latent representations, and we can do linear interpolation between them. And what's interesting to see is what's going on in between, because that helps us to understand how this network works. So what I show here is the one structure, the other structure, and then this vector represents the vector features. So this was a different type of architecture where we just tried to compress into a very small latent space with 1 by 1 by 1 grid. So it's a voxel of like small size, and then it also has vector features. And this rotation just shows that the it's the interpolation. It doesn't show the equivariance here. Okay, so next. Okay, so to sum up, so in small fragment binding free energy prediction, so we have seen that model has learned basic features of frag maps, meaning protein surface versus core, or overall occurrences of binding hotspots. So AC3 cognate, they don't need to learn orientations of different motifs. So in order to make these predictions more accurate, we need more data. So this became a grant proposal. And in the future, we expect more data, and we are recently applying new architectures. So with another project, which is learning new structural representations. So this unsupervised compression achieved 97% accuracy. Equivariance is better compared to this, general neural networks, convolutional. So in the future, we are trying to also do a different direction in the model, which is called atom typing, and this kind of models we derive from language translations model. For example, these representations of the densities can be represented in different ways. So, okay, so I'd like to take a moment to thank all my group members in Lamaro Lab. These are the people, and thanks for your attention. Any questions, please don't hesitate.
Thank you very much for such an interesting talk, Dr. Talant Uzumetov. Really, reducing simulation time from 4 days to 1 second, it's really an amazing result. I can say that this is the main reason that scientists are concentrating on applying the artificial intelligence to solve complicated problems in the science. So I don't see any questions. Thank you very much one more time for your talk, Talant. Now, next in the line is Dr. Shaker Pardaev. He is the chief engineer in Intel Corporation. He provides technological solutions through photolithographic process development and critical analysis in a semiconductor manufacturing environment. Optical lithography is the standard process of printing the pattern of an optical mask onto the surface of silicon wafer to manufacture microprocessors. He also responsible for developing the lithographic patterning solutions to transfer the final pattern on silicon wafer. Lithography is not only a technically challenging process in the semiconductor manufacturing, it's also often the limiting factor in entering the development of manufacturing of next technology node. So he obtained his master's degree at Mississippi State University in 2009 and obtained his PhD degree at Kent State, Ohio University in 2017. Please, Dr. Shaker Pardaev, you can start your presentation.
Okay, good morning. Can you hear me?
Yes, yes.
Hey, good morning, everyone. So as Talant was mentioning before, we are 3 hours behind. It's just, it's not even 6 o'clock in the morning.
Oh.
I had to wake up at 4:30. I'm sleepy, so if you just have questions, you can keep it then.
Okay.
So, you know, I gave the option to Jamal what kind of topic I should be speaking in, to make it more technical or to make it more general, like a story. And since this is the feeling of a conference, which is problems on modern science and technology, I thought maybe I can talk about my research that I did when I was in the graduate school. So can you see my screen?
Yes. No, it's okay.
So during my graduate school at Kent State, I worked on the soft matters. It was mainly on liquid crystals. Liquid crystals, they are used everywhere, starting from your iPhone or tablets or big LCD screens or the computer monitors. All of the screens, monitors like laptop monitors that you can imagine, they all use LCDs. But now they are slowly transitioning to the LEDs. So I did a few projects during this graduate studies, it was like 4 years ago. It was mainly, we studied different defects in the liquid crystals using the light scattering. mostly using the second harmonic generation, which is you send the laser light with one kind of wavelength and you get double wavelengths. And why it's interesting, it's not only a matter of generating another wavelengths by the signal of the secondary frequency, we can extract that different properties of the matter that we study. So the talk, I will try to make it short. It's so there were two parts. So I'm going to talk on the one. The first was the second harmonic light scattering of the topological defects in liquid crystals, and I'll give a little bit of introduction of the why we do and what we do, and some experimental details and some results. And if there are time allows, then I can talk some about Intel and my current job. So the liquid crystal molecules, they usually, initially, it was kind of rod-like molecules, which is dipole moment, 10 dBi, and they're usually polar. But when we just, liquid crystals, when we just align them together, liquid crystals, they are like the phase in between the liquid and the crystal. When you heat up the temperature of a material, then it goes from crystalline solid to the liquid, liquid crystal phase, and after that it's a liquid phase. So when it takes, if we just take some average of this dipole moments of this alkane, that this represent one molecule, they tend to cancel the polarizations. This is a nematic liquid crystal, and it's a centrosymmetric where all the polarizations, they cancel each other. But when you apply some magnetic field in the liquid crystal, they form some sort of walls, and we call it inversion walls. And where the polarization, there is an induced polarization, and it doesn't cancel, and it breaks the centrosymmetry. And along this walls, we see some defects form. And this is different kind of molecule. It's kind of two rods connected with a flexible chain, and they form different kind of phase, which is it's nearly fairly newly discovered phase in the liquid crystalline area, which is it's called the twist-bend nematic phase, and it was a big kind of discovery. It's a forced phase in the liquid crystal environment, besides the nematic, the discotic and the smectic, and this one is a twist-bend nematic. So they are also polar structures, and when we apply the magnetic field to the this phase, and they also form this kind of defects, and we call it the parabolic focal conics. So we basically in the research we did study this inertial wall loops using the magnetic fields in the second harmonic generation, and also parabolic focal conic defects. So using the second harmonic generation, this is a very good technique to study the polar structures. So in the second harmonic generation, what's this? So usually the in conventional linear optics, the polarization is directly proportional to the first power of the electric field. But in the second harmonic generation, and in the nonlinear optics, it's proportional to the second power and third power electric field, and so on and so on. And we only consider it on the second power because that's when the centrosymmetry of the structure is broken. So if it's not centrosymmetric, then it's basically zero. So in the nonlinear medium, we send the laser light with the frequency omega. Then it goes after this nonlinear medium. Then you see the first harmonic and the second harmonic, and by some filter we filter out the other harmonics to only filter the first generation. So there are some equations. So we studied in two different kind of cells, which is, the first one is the 1 millimeter cell, is the 100 micron cell to study the thickness dependence, how these defects are forming. This is a setup where I used. and developed and built this. This is a telescope, and the main part is the magnetic field and laser. It's the sample sits here, and this Indiac laser. This camera is very sensitive to even a single photon, and this one is a telescope that maps all the scattered light to the camera. And that's how it maps. So the sample and just goes, and by this the telescope, it just gets mapped to the camera. So some results. So in the first part, we studied 3 kind of different molecules. With the purpose of the first molecule is a polar molecule. This is polar. This is a non-polar molecule. The last one, and this has a different phase, and around this temperature, it shows the isotropic, nematic, and the crystal, and at different phases. And this is the shape is different, so we expect them to deform at different kind of phases. So then we heat up the isotropic phase and cool down to the nematic, and where you in the magnetic field, and we see different kind of defects. Where I showed in the earlier presentation, earlier slides, that they are kind of inversion wall loops. And we try to then we shoot like the lasers in the sample and see what it gives. And this is basically in different kind of cells. This is 1 millimeter, 1 millimeter, and this is 10 times smaller, it's like 100 micron cell, where the defect, the number of defect density is much less than the 1 millimeter cell. So these are the typical second harmonic scattered light. Then we see the 2 peaks, and these 2 peaks they are separated by some distance, and you can see that as we cool down the temperature, and the peaks are getting bigger, which is the defect size is getting smaller, and that's scattering like more light in different angles. And we can approximately see the size of the defect, which gives around like 3.2 micron. And this is from different materials, that bed core and 2 rod-like molecules. They all have a different, they all have a similar shape, similar features, where they're just like in a larger angle. Basically, it says that it's the defect size is smaller. So you can see that, as I said, these 2 peaks are as we cool down the temperature, and they're getting bigger and bigger. So the separation is getting bigger. And this basically in the liquid crystal, there is an order parameter. So this order parameter is basically is getting more order. So by cooling down the temperature. And we try to explain that this second harmonic generation pattern by this Landau-Dijon free energy, and we model, and we try to see if it gives a polarization that we expect. And we see that this after solving and minimizing the free energy, we see these 2 polarizations, and you can see that in the top and the bottom, or this range, they cancel each other. But on the left and the right, I don't know if you guys can see this 2 polarization, they kind of break the symmetry from, you can see one is pointing up, one is pointing down. So basically breaking breaking the center symmetry, so it's not non-center symmetric, so it's generating the second harmonic light. So the second part is different kind of molecule. So which is the rod-like molecule. It's connected by the flexible chain, and they form. We basically studied this molecule. It shows the isotropic, nematic, twist bend, and smectic X, and the crystal after some time. And this shows a different kind of defects. This defect is a representative of the focal conic defects. And basically, as we cool down as we go from 1 minute to 100 micron, where the defect size is the space where the defects can form, they are much smaller, so that there are less defects on the 2 surface. So this is a representative of the signal, where resolution of the signal was not that easy. And as we is in different geometry of incident laser and different polarization. So we see again two different kind of peaks, and we try to account for this by there are two peaks same, and there is some noise, and we could not really reduce the noise because of the intensity of scattered light was not great, we thought. So it follows all the procedures and follows, it doesn't break any rules. And this is of parabolic focal conic defects, and we try to develop a model where we can explain this scattered light too, and we use the Landau division free energy with different kind of terms. These are the polarization terms, and then we try to minimize this free energy using in terms of the polarization. So these are the pneumatic field and the polarization field, and we basically plug all of them into this complex free energy and minimize it. And at the end, we get this two components of the polarization, where it's in the same direction where we get the generation of the second harmonic light. So it also shows the ratio of this two polarization, where it clearly explains the signal that we observe. So in the summary, in the first part, we did this in the magnetic second harmonic light scattering on the magnetically aligned samples. We try to relate this angular distribution of the peaks observed in the scattering pattern to the presence of the inversion wall loops. And there are different kind of terminologies, which one of them is flexoelectric coefficient. We try to deduce the approximate value for them. And the second part is this second harmonic generation technique. We try to investigate the molecular organization and then pneumatic twist and phase. And we observed this focal conic defects. Yeah, so this is it. I stop here. I don't know how much time it took.
Yeah, it's okay.
30 minutes or 13?
You have a couple of minutes.
Okay, if I have a couple of minutes, if there is an interest, then I can talk something about the Intel and
Yes, please.
Yeah. So this is after finishing PhD, and I joined Intel Corporation. It has many sites, and the main development site is in the US, in the US and Oregon side, where I started. And I work in the lithography department, which is like completely, it's kind of similar to the science, but it's completely different from what I did in the PhD. But we use mostly optics, but they use the skills. So this is Intel Oregon, and Oregon is on the west coast of the U.S. And then next to the Pacific Ocean. And it's really beautiful. It has a really beautiful nature, and it has a lot of falls and forest and volcanic areas and ocean beach. So Intel, it was founded in 1968. So it was founded by two graduate students, two PhD students from Caltech, California Institute of Technology. So they basically got Robert Noyce and Gordon Moore. They were an employee engineer in one company, and they had some sort of, I think, the problem with the boss. So they had to leave, and they started the company by themselves. And the first chip that they made was in 1971, which is the Intel 4004. It was the first commercial available microprocessor. And 1974, there's a different x86 architecture came out. So since then, like, you know, the Intel has established a heritage of innovation that continues to expand the reach and promise of computing while advancing the ways people work and live worldwide. And whatever computer chip that you open, it has an Intel microprocessor. And right now in the world, there are like more than 100,000, like more than 100. It's still 111,000 people. It has like 190 different sites in 90 plus different countries. So what there is in semiconductor industry, there is a law. It's called the Moore's law. Basically, what it says, integrated circuits every year or every 2 years. So the number of transistors in a chip doubles approximately every 2 years or every year and every generation. So basically, it cuts the cost while increasing the performance. So decreasing relative cost and an exponential at the exponential pace, it increases the performance. So initially, it started like the micron size, then 130 nanometer, 90 nanometer, and 65, 45, 32, 22. And when I joined Intel, we worked on 14 nanometer. Now it's 10 nanometer, 7 nanometer, 5 nanometer. There is a huge race going on, like how much you can make it smaller. And a lot of people are predicting, okay, it's the end of the Moore's law, so you cannot just make the transistor even smaller. But there are different kind of techniques that the technology is using. So Intel is investing a lot in the infrastructure, and only 1 quarter earning was $20 billion, which was like 20 in Uzbek, $20 million. So in the only R&D in the 2020, that $28 billion was invested in the R&D, only R&D, and the total earning in the 2020 was almost $78 billion. It's way more than the GDP of many countries. So this is Intel is investing a lot into building new factories and new development. So a lot of people has been predicting, like, okay, there's the Moore's law, so we cannot make the chips even more smaller. So that was when in the very first 1899, where the U.S. U.S. Patent Commissioner, they predicted, okay, everything that can be invented has been invented, so no more. So then in 1988, after like 20 years after the Moore's law won't work at feature size less than 1/4 of micron. Okay. And decades later, in 2010, a lot of people predicted life after Moore's law. What's going to happen after we cannot reduce the transistor size anymore? And in 2009 and 2016, Moore's law is dead. And what now? What we're going to do? But so if you just look at the history, the Moore's law was dead almost from like 300 to 400 times. So the people predicted, okay, no more. But there is technology just keeps coming. So how Moore's law can be kept alive is just invent, and new nodes. So how are you gonna do it? You're gonna change the architecture. You're gonna change the materials. You're gonna change the architecture, materials, and new materials, new architecture. So the transistor size right now, the 7 nanometer. A 7 nanometer, if this is a DNA size, right? DNA is a 2.5 nanometer. There's a 5 nanometer kind of vial filled with water, right? So the 7 nanometer is like around 3 times more than the DLA size. That's how much stuff that we can print on a transistor and on a wafer to make a chip. So now is a 3D stacking and plus the 3D stacking of the transistors, and there is a different packaging. That packaging that makes a huge difference. So different kind of chips and a package and make a different strong chip that can be used in the artificial intelligence computers. And it's also very low power and so as in what happens in the development side, right? There is a pathfinding, so they come up with idea and the architecture design, then equipment development and yield and manufacturing. Like so, our job is like a cycle, like pathfinding, just find the new ways to do to implement, and the design architecture, the designers, and the equipments. All the equipments are very clean in a clean environment. So, all the people inside, they're kind of they're experts inside the fab. Fab is very clean environment. So currently, because of the pandemic, right, and there is a huge shortage in the chip, and a lot of car factories cannot make cannot produce their cars because of the chip shortage, and the Intel chips are basically now used in the AI analytics, high performance computing, and a lot of cloud-based and data centers and the networking, database, virtualization, and security. So that's it. So if you guys have questions, please feel free, and I will try to answer.
Thank you very much for such an interesting talk, and that this presentation was quite informative about Intel and processing the chips and making it small and small. So I don't see any questions. I would like to thank Dr. Pardaev. Despite the inconvenient time, you managed giving a talk in this conference, and we wish you success on your professional activity. So thank you one more time.
Okay, yeah, thank you. Thank you, everyone. Have a good day. I mean, have a good evening.
Yeah,
You're gonna have a good day.
Mm-hmm. Yes,
Good
sure.
job. Mhm.
Right. So we would like to say thanks again to presenters for the informative and interesting talk, and to audience for active participation. Hopefully the presentations will be beneficial for everybody. And thank you for your attention, and thank for who took part in organization of this conference, especially for translators as well. So the following sessions will continue on 11th of July, tomorrow. So agricultural engineering, energetics, civil engineering, advanced chemical technologies, and computer science and technology. Thank you one more time for your attention and participation. See you tomorrow. Have a nice evening.